this is cool. Now we have discovered something very, very unique, a non-apoptotic cell death inducers that is killing the cell through non-apoptotic mechanism. And in colon cancer, if you see, most of the drug that is used kill the cell through apoptosis, a mediated cell death mechanism. So one question we ask is, can it overcome apoptosis resistance, or will it be effective when we give it with other anti-cancer agent? So first thing we wanted to show that methodology is independent of back. We created a single and double knockout models of colon cancer cells. And when we gave BAPT, it killed all the cells at the same concentration, while 5-fluorouracil, which is given in colon cancer, was highly resistant. You have to give a lot of drug to kill the cells in double mutant line. Here, they will kill at the same concentration, right? Which suggests that it can bypass apoptosis resistance. So we started combining it with different anti-cancer agents. And we found that BAP compounds were highly synergistic when given in combination with other anti-cancer agents. So it can be a very good adjuvant when combined with other anti-cancer agents. It is pretty safe when you give in cell lines. So as I said, it was a good anti-cancer agent. We always have to do toxicity studies. So we looked into normal cells, fibroblast cells, and it did not kill at a very, very high concentration. In mouse models, we did not see any change, significant change in body weight, or no apparent toxicity or behavioral toxicity was observed. In normal colon cells, we did not see even vacuole formation up to 30 micromolar. There was no vacuole formation. Including zebrafish, we did not see any significant toxicity. Overall, we found that this compound was pretty safe in killing colon cancer cells, but does not have other adverse effect at the concentration that we tested. We also looked at organ toxicity in different organs. We did not see any significant change. We looked at kidney and liver function. There was not much change, and even the blood chemistry did not show any significant change, which was very, very interesting finding for us. And then we developed some patient-derived xenograft models to study efficacy studies, and which we are doing right now. One question that becomes, and you may be wondering, that looks like a very interesting cell death mechanism, non-apoptotic. The drugs are effective. It can overcome, apoptosis resistant. How is it working? What is the mechanism? And this is the kind of question that we ask. Okay, this is cool, but how is it doing what it is doing? So, like most of us, we do omic studies. We did omic studies to find out what are the targets. And of course, metabolic pathways were highly dysregulated. Not going to go much in detail in this, but metabolic pathways were highly regulated. We then also did kinome scan to find out which kinomes were affected. The original compound was multi-targeted agent, as you can see, but some targets were very clear. Like PIK5 was very, very clear target. Junk pathway was very clear. That was highly, inhibiting at a very, very low concentration, which was very, very exciting to us. Um, and then we further followed it by looking at the lysosomes, how the lysosomal biogenesis and membranes were being affected. And we found that lysosomes were not releasing these autophagosomes outside, and they were highly acidic in nature. So it has to do with a signaling pathway that affects lysosome to nucleus signaling pathway, which is mTOR TFAB. And so clearly our compound was inhibiting TFAB signaling pathway significantly, which suggests that it is actually targeting mTOR TFAB signaling pathway. So in addition to PIK5, mTOR TFAB signaling pathway was our key target for these class of compound. So to summarize, what we found so far is that in colon cancer cells or colorectal cancer cells, when cells undergo autophagy as a normal process because they need nutrients and metabolites for cell growth. But when you give these BAPT compounds that we discovered, they form these vacuoles, also called methoosis. There's a lot of accumulation of RAB7, LAMP1, RAB5. At the same time, this autophagy is inhibited, right? In fact, autophagy is dysregulated and there's too much of lysosomal disturbance because of mTOR TFAP signaling pathway. Okay, so there's something going on here. At the same time, this methuosis and autophagy is both leading to cell death. Okay, so that's what working model for methuophagy we have so far. Since then, we have made a huge progress in doing structure activity relationship to identify more novel compounds and synthesize a large library of compounds. As I said, we have now three patents, US patents on this technology, and we have covered significant chemistry space in this area. We have also identified a different structure activity relationship to identify which compounds will do lysosomal to nucleus trafficking and how distinct they are. In doing that, we can tell which compounds will only do methuosis, only do autophagy, or can do both methuophagy. So we have a lot of handle on different type of cell death features and chemistry space in this area. Very exciting work, and we continue to develop this further. We have many other technologies that we have been working on, as I shared earlier. One quickly that I want to run in next 2 or 3 minutes, and then I'll wrap up my presentation, is another technology that we found in triple negative breast cancer, which is a very difficult cancer to treat because it does not express the key targeted factors such as HER2, estrogen, and progesterone receptors. And the only treatment that works initially in these patients are taxanes and anthracyclines like paclitaxel, docetaxel, doxorubicin, epirubicin, and so on. But they become resistant because of overexpression of these ABC transporter, DNA damage repair, or generation of these ROS, basically apoptosis defects, which leads to metastasis. What we found is a new class of compound called TPH104, which is highly effective. It blocks these efflux transporters. It inhibits DRP1 protein, which is a dynamin related protein 1, which leads to mitochondrial fission inhibition, does not go through apoptosis, so it's a non-apoptotic cell death. It leads to a necroptosis cell death induction. So that's why I was saying this is the first small molecule to induce necroptosis through RIPK1 MLKL pathway. This is immunogenic in nature, meaning it activates our own immune system. Not only it's killing the cancer cells, but it is activating your own immune system to further, tackle other cancer cells. So it's a very powerful system that we have been working on and developing, and we just recently got another US patent on this one, which is very exciting. And it was a huge discovery for this class of compound. In fact, we developed a 3D lensless holographic microscope through machine learning and AI, and we can now predict different types of cell death phenotype. So it's a very small $300 microscope, very cheap, like 10, 15,000, 20,000 rupees microscope that is AI and machine learning configured. It can tell you when you treat the cells whether the cell is going through apoptosis, necroptosis, pyroptosis, or methuophagy, or different types of cell death. Through that, we actually discovered this TPH pharmacophore to begin with, and we have optimized to a very, very, very potent compound up to nanomolar concentration, which is very effective and very selective in specific cancer like triple negative breast cancer compared to normal cells, up to 150 to 500 fold selective. And we have developed new models of drug resistance, and we can show that it completely eradicates drug resistance. I have a ton of data on this project, and maybe we can find another session next time to talk about this very, very interesting project. But I will have to wrap up because I think I'll otherwise go over time. And I'm really always thankful to my team who's doing all this work and allowing me to present this work to you. And again, this is my team at UAMS. We have 6 PhD students and 4 research staff, which are very, very actively working on these very exciting areas. And I really want to thank funding agencies which support these work. And also thank you, Dr. Dahiya and APP and other leaders who are here, and of course students who are present today to attend this session. I'm open to questions. If you have any questions, I'll be happy to take some questions if we have time for questions. But if you're running out of time, you can look me up at LinkedIn, connect me there, ask questions. I'll be happy to answer any other questions there as well. All right, I think I'm pretty much done here.
Thank you, Professor Tiwari, for sharing and educating us with your research. And I think the lecture was wonderful and it will be helpful in many research projects. And they will, student will be in touch with you. If any specific question is there, I want to ask anybody.
I have a
And
question, please.
yeah,
Yeah,
Professor.
yeah. Thanks, Dr. Tiwari. Very nice presentation and nice work. I have I have one question at least to ask. This difference between methodophagy and the autophagy. This methodophagy can be considered for lung cancer targeting model. This,
Yeah,
yeah.
yeah, good question. Good question. Um, Autophagy is part of methoophagy, meaning these compounds are also doing autophagy and also doing methoosis. So there is that, right? So autophagy is also being induced. So what we have found,
Yeah.
these drugs are very effective, extremely effective in GI cancers, right? Pancreatic, colon, some RAS-driven cancers, glioblastomas, very effective. Non-small cell lung cancer, we have seen some effect, but I would say that it is not as promising as what we have seen in some other cancer.
Because you are working on cancer, just one doubt I want. This non-small cell carcinoma and small cell carcinoma, particularly lung targeting, right? Which one will be better model? Means, most evident.
Yeah, non-small cell lung cancer is much more easier.
Because
And
one of my scholar going to work on that, so we can target non-small cell carcinoma in lung targeting, right?
right,
And
right.
this autophagy, methodophagy techniques by utilizing saying paclitaxel or its derivative will be effective.
So yeah, you can combine methoophagy with any anti-cancer compound that you want. But yes, when we combine with taxanes, paclitaxels, and other thing, it was very effective. So it is
So
synergistic.
Taxol will be best model, best drug for that,
Yeah.
drug of
So,
choice.
so yes, Taxol is fine. We also have Taxol resistant models, and it
Yeah.
works very well in Taxol resistant model as well. So yes, Taxol is fine.
Okay. If any help required, I'll communicate you over phone,
No,
over the
no,
mail. So
no
we need your
problem.
assistance in future. Thank you. Thank you for a nice
No problem.
presentation.
Yes.
Yeah, this is Shanmu Sundaram, one of the chairperson, and thanks to Dagya sir for giving this opportunity, and thanks to Amit Tiwari. It was an excellent presentation. I don't have any specific questions, but the oncology session in the morning was quite good. Thank you, Amit. Thank you.
All right.
Good morning, sir.
Good morning.
Sir, I have one doubt that you have discussed that defective apoptosis is the one of the major cause of the multi-drug resistance. But in case of defective apoptosis, they are present mainly two factors. One is extrinsic factor and another one is intrinsic factor. Sir, in case of extrinsic factor, I have read that the death cell receptors are the main part of the extrinsic factor. But I am not clear about the death cell receptor, how it affect these defective apoptosis. Can you discuss with that?
So extrinsic and intrinsic apoptosis is only mediated by where the signaling is coming from, right? If it is inside the cells, it's intrinsic. If it is extracellular signaling pathway, it's extrinsic. And they all merge into execution pathway of causing apoptosis. Ultimately, it leads to, you know, ending up into the same machinery. So no matter where the mutation is, okay, these non-apoptotic cell death inducers should be working in those process. And it's very difficult to address like which signaling pathway, which mutation would lead to, triggering or making apoptosis ineffective. It's very hard to pinpoint because different drugs have different targets, and when they work, they get resistant because of different factors. But ultimately they stop responding, and you have to switch another drug and another drug and another drug and another drug, but yet you want all of these other drug to kill through apoptosis. So what we are trying to accomplish is bypassing this apoptotic machinery completely and then killing the cells, which may look very simple from the surface, but really most of these cell pathways are interconnected, meaning there are very clear link between apoptosis and some of these other cell death like pyroptosis, necroptosis, mitosis, entosis, panoptosis. So there are very subtle differences in that, and we are all figuring it out right now. The research is only recent. Some of the cell death processes are being discovered every year. For example, panoptosis was discovered just last year. So we are learning a lot. So to your question between extrinsic and intrinsic, it's the mutation can happen anywhere, whether it's external factors or intrinsic factors. But ultimately, if there is a resistance, you can overcome with non-apoptotic cell death induction. Should be in theory.
Okay, sir. Thank you, sir, very much.
Yep. All right. So thank you so
Thank
much,
you.
everybody. And yeah, it was exciting to talk to all of you.
Thank
Yeah,
you,
if you can
Professor
connect me on
Tiwari, for
LinkedIn
your
and
time.
yeah, and then people can connect me on LinkedIn. I'm usually very responsive to help.
Yeah. Thank you, Professor. So, uh, in continuation of the lecture, now we invite Professor Wang to deliver his talk. Professor Wang, you can continue your talk. Thank
Okay,
you.
thank you. Thank you. Thank you. Very nice talking. Very nice talking. So, first of all, I thank you, the president and the committees of the APP. So, invite me to come to this conference. So, this very nice talking. So, today I want to talk about the gut microbiota. So, as an invisible organ. Previously, we don't think the gut microbiota is an organ, and this how can influence our body, and particularly hepatic diseases, means liver. And first, I will introduce myself briefly. I graduated in China. My major is Oriental medicine, so I'm a clinic major. But when I have a chance, I came to the Korea and finished my master's degree and a PhD. From 2004, I started to do the research work is related about the liver, the hepatic and the metabolic diseases. And when the 2013, and I come to the Donggu University, and I start to research for the regarding the gut microbiota. And now I work in the Donggu University as a research professor. So as everybody think like this, every researchers do the researchers like this. When he does the bachelor degree, master degree, and a PhD, he said he work, study hard, and think hard, and do the research hard. But when he finished, he do the research, he find a lot of things we need to do, we need to think. It's really not easy work. So I give some tip. So when we do this, we just found the small fragment and make this the small fragment together, and to see the iceberg. And when you see the a lot of the news and including the FDA also approved some the fecal microbiota products coming. They can treat some diseases, and also fecal microbiota capsules coming, approved by FDA, and also like Jackson Laboratory, they said the gut microbiota maybe they can predict some the fatigue syndrome and the long COVID like this, and also some place in the famous journal also they said oh the drug and the microbiota they can interaction and change the drug effect. So maybe you think oh this maybe some effect some they can do some making they can work and they can impact on our body. So previously we always think about the pathogenic the bacteria like some Salmonella and the E. coli and some the microbacterium, the tuberculosis. Like this is almost can induce our body the injury, like a lung injury and infecting injury, means the inflammation, serious inflammation. Also, they can induce some the sepsis and diarrhea and some the intestine problem, constipation. So but recently, we don't focus on this pathogenic bacteria. More and more studies focus on this other bacteria. That means the beneficial bacteria or the actually the opportunistic bacteria. That's we don't know this is good or bad. Sometimes they come back good things. They do the good things. Sometimes they do the bad things. They can do that is sometimes it's out of thinking. They can do a lot of things out of our thinking. They can promotion the fat storage. They can promoting the angiogenesis. They also the training the immune system. They also can break down or make the vitamins or amino acid. They also can interact with the other the organs, the like neural system, and this actually can do a lot of the works. But this bacteria, if these bacteria are unbalanced, that means dysbiosis are this the microbiota. So through uh, some types of the means gut brain access or gut lung access, gut skin access, a lot of the access they can make our disease, make the disease. Uh, so that's the reason. So not only government, all of the company will invest a huge money to do this research. For example, the USA government launched together with the government, together with the company, a lot of the company, they launched a national microbiome initiative. That's investing the 780 million US dollars within 10 years. So there's a huge investigation, huge the research funding for the microbiome research. If we see if they see the academic publications, we also see from in the 2000 years, only 5 papers together with the gut microbiota. But after 20 years later, there's more than 100 times the publication has increased, significantly increased within 10 years in the worldwide. And within this publication, let's see that they have the different area, the most of the area, but the biggest area is the gastroenterology and hepatology. That means that the stomach or the stomach intestine or the liver. So today I want to talk about the gut microbiota and some liver disease like alcoholic liver disease, injury, and the liver cancer, or the non-alcoholic fatty liver diseases. So I heard this some students will come to here, so I add some background knowledge of gut micro. I think maybe some students they don't have the knowledge, some basic knowledge of the microbiology. So I add some the background knowledge inside. So first I will explain some background knowledge, and then I show some the evidence of the gut microbiota and the different the liver diseases. And finally, our summary to the how to use this target on the gut microbiota to in the future to make some do the application to do the drug development. So First, what is microbiota? So we think microbiota is the microorganisms. So we cannot see this living in our gut, and the human or animals living that. But actually, they're not a bacteria. Not only a bacteria, they also including the archaea, fungi, or virus. They are the common the characters is we cannot see that it's very small. And previously, we always use the gut flora, but now we use the gut microbiota and the gut microbiome. So after that, I will tell you which the different. Later, I will tell you what is the difference of the microbiota and the microbiome. A lot of person when he write paper, they not distinguished. They just use together with they use together, but actually it's totally different. So I attend some conference. One professor in Korea, they found kimchi is a traditional fermented food in Korea, very famous, the kimchi. So most of scientists they research a lot of the lactic acid, the bacteria. That means the lactobacillus in the in the kimchi fermented food. But actually, that one Korean scientist they found only in the kimchi they find a lot of the virus. That means this commercial bacteriophages means in the surface of the lactobacillus, the bacteria surface, they found a lot of the bacteriophages in there. So they said this is not we don't worry about that. So maybe this is together with us for the thousands of years. This may be the safe for ours, but we don't know. I also think this is opportunistic. When some situation will change, the environment will change, and this is the bacteriophages also they can change the lactobacillus and also impact on give us the effect to us. So and next is I want to explain what is different from microbiota and the microbiome. So a microbiota is only the structure, is only the community of the microorganisms. So most of the paper, they only mentioned the bacteria, not mentioned for other things. That's just the community of the microorganism. But microbiome is the more wide area that's include not only the microbiota itself, they also consisted the metabolite, including the DNA, RNA, the lipid, and also the polysaccharide. Make this together, all of things related with the microbiota, related with microbiota, they also combine together with the microbiome. So microbiome is more. If you only do the 16S sequencing, only check the structure. Only that means easily to say that if you only check the structure, which bacteria inside, which bacteria in there, this is microbiota. But if you checked how the microbiome do, what the work, what they do, how to do, that is microbiome. This combined all of the elements. That means microbiome, the structure elements. So and bacteria, we don't in normal people we don't know the bacteria how big or how small is more big than human cells or small than human cells. Through this picture, you can say this is our blood. The red blood cells is about 7 micrometers, but there's bacteria is even small, from 0.5 to 5 micrometers. It's really small. That's the reason when we do the culture cells, why we use the 0.45 micrometers the syringe to filter the bacteria. So that's a filter. The syringe filter cannot filter the bacteriophages or viruses. It's impossible. So we also difficult to through the optic microscope to see the bacteria. It's very difficult. If we want to see the clearly, so we only we can use the confocal microphone or electron microscopes. Yeah, and same as a human, bacteria also they have the through the taxonomy, from the phylum, class, order, family, genus, or species also like humans. So the total is total 50 fire of the bacteria in the world, in the whole world, in the only 50. Oh, sorry, in the only 50. But in the human, got different person and different areas different. Only 8 to 12 fire in the human gut. And most big is Firmicutes and Bacteroidetes. Is about the 90% of bacteria is belong to the Bacteroidetes and Firmicutes. That's both of the two. And regarding to the species, around 500 to the 1,000 bacteria species in the human's gut, the intestine tract. So one famous professor, they write the one book is Missing Microbes, is from the Martin Blasser. You can search, read the book. It's very interesting. So that means from the previous last century and through the long times, the modern society changed, and our microbiome, the bacteria the species is continuously significantly reduced. They will reduce far and far. That's the reason a lot of metabolic diseases will happen, like the diabetes and obesity, like this, will totally change. This bacteria is very important, the function in our body. And yeah. So through the research, previously we don't know that because of the no instrument to see that is too small. The first the Leeuwenhoek, so they found they developed the microscope. They found firstly they found this is a father of the microbiology. They found the bacteria, microorganisms, and following the technical development, a lot of the machines, the sequencing technology, sequencing machines coming, the Sanger sequencing, the first generation, second generation, the third generation also already we can use. Now we can easily and very fastly to distinguish which bacteria inside and who is there, which bacteria in there, and what they are doing, and how they are doing. That we can distinguish the bacteria, gut bacteria, in very short time, and very in one time we can check all of the bacteria through the sequencing technology. So first time is more popular, and we use the 16, the ribosome RNA to distinguish the microbiota, the bacteria. The reason is almost the bacteria species, they have the 16S rRNA. So we use this 16 RNA to do the sequencing. And this is the 16 RNA genes is highly conserved. And if they are too short, the information will be less. If they are too long, maybe it's difficult to do the sequencing. So that's the reason 16 RNA, 16S rRNA is the best choice for the development. They have the wiring regions from V1 to V9. We distinguished to check the distinguished identify the bacteria. And another method is the shotgun, the full genome sequencing. We can know, we not only who in there, we also know what they do, how they do. But the shortcoming is too expensive. Now is they reduced too much, but it also is not cheap for the detection the bacteria. And we also, when we do the research, we also use two methods to delete the bacteria. So one is antibiotics to delete, and other is the germ-free conditions. Use the germ-free conditions like the isolator to maintain the germ-free environment. And as we know, the last century we used in the very famous project is a human genome project. We detect all of the genes in the human genes, about 30,000 genes in human, in all of the human. In that time, we can say, oh, we can know all of the diseases, the reason of disease. But actually not. Until now, we know that we are not an individual. We are is an ecosystem. Together with us, a lot of bacteria is also in there. So that's the reason the Nobel Prize winner, the Joshua Lidberg from USA, they said, we are not a single exist. We are supraorganism. That means together with the bacteria, we are living together. is a supernuclear. And also in the from the west, from the east, east and the west, the scientists also say the similar things like that. In the Oriental, means the West scientists, we thousand years ago, we also said this word, the fecal toxins. It means in the fecal toxin can come into the bloodstream and they can lead to the a lot of diseases. And in the Western medicine, the father of the modern medicine is Hippocrates. They also said all the diseases begin in the gut. I think so. That's the reason we also think of questions. Maybe in the gut, maybe something happened where they are infect our the body. They do some works. So today I talk about the liver. So as we know, the hepatocyte is they can make the bioduct and they the bioacid and through the they make the bioacid and through the bioduct they secrete to the intestine and to the previously we think that is the only digest the fat, but actually we know that it's not for that. They also modulation the bacteria and do a lot of more the properties. So and after that, a lot of things they do that, and a lot of microbiota, the metabolites, and some the cytokines, and the and we'll come back. We'll go back through the mucosa and go back through the mucosa and come to the photobain and go back to the liver, and that is the circulation. We said is the enterohepatic circulation, and this in fact to our body. So first we are talk about alcoholic liver disease and gut microbiome. So maybe in India also a lot of people can like drinking the beers or some alcohol. So it's very easy. Previously, we always think, oh, this is I can drink very much because my liver can secrete, can make a lot of the enzymes. They can break down very easily, very fast. I can say strong. They can easily make it to the acetaldehyde and acetate. So and come to the circulation. We always think like this. Oh, I'm a strong. But do you think about that? When we drink the alcohol is not first they come to not directly come to the liver to metabolize because alcohol is a metabolite to the liver. Most of the alcohol, they come to the stomach and come to the intestine, and then they observed to the blood. So maybe before they metabolite metabolized in the liver, they may be together with the stomach or the intestine. There is something will be happened. So that's the question. So does the alcohol can change the microbiota and the gut microbiota? The microbiota in the gut is changed. So a lot of 10 human studies showed after you drinking, they must be changed. They change. Some bacteria will decrease, some bacteria, the abundance will be increased. So a lot of change. And also the structure, the total structure, the yellow, the person and the blue person. Blue person is the blue one is drinked. They are also totally different. The structure of the gut microbiota. So that means, so alcohol can change our microbiota. That is alpha diversity. Alpha diversity means how many species, like for that is the index, the Shano index, the Evanis. This showed the numbers of the bacteria is both of drinking. After drinking, they are decreased. The significantly decreased. So not only in the adult, in the children also they are decreased. After drinking, they are but if you're not drinking, so maybe they can slightly recover the alpha diversity of the microbiota of the biota. And also another study showed after we drinking, the 16 RNA exists in the all of the bacteria in our serum, in our blood. We found the significant high, the higher bacteria after drinking alcohol. That means our the blood, our the intestine, the intestine permeability will increase. So a lot of things they are increased the permeability. So a lot of the metabolite or the bacteria, so we are through the portal vein come to the liver, and induce the immune system activity. So one another study also showed after drinking only in the 30 minutes your endotoxin, I always we check the endotoxin. Endotoxin will increase very fast. This endotoxin through the portal vein and come to the liver and activated the Kupffer cells and Kupffer cells to secretion the inflammatory cytokine and make the liver injury. So that is another reason of the alcoholic liver injury. They're very fast to increase. And interesting, the woman is more higher, woman is more higher than men. So that's the reason the woman is weak is weak than compared the protect ability is weak than men. That's maybe the endotoxin is one of this reason. So. And another is the time junction proteins. This is detect the permeability of the gut. You can see this very strong in the control, and after you drink, all of these proteins, connected proteins, will reduce the significance, reduced, but interesting, what if you eat the butyrate. Butyrate is one of type of the short chain fatty acid. Short chain fatty acid is the metabolite of the gut microbiota. Gut microbiota, the bacteria will make the butyric. Butyric will can protect your gut permeability, your gut the integrity. So this is a small, the brief summary of the alcohol liver disease. So if you drink alcohol, your gut microbiota will decrease, will dysbiosis will change, and also they reduce the alpha diversity, and also they can increase the LPS, the endotoxin, and also they can decrease the short chain fatty acid like butyrate, and also enlarge the permeability, the gut permeability, induce the inflammation. And the human is because of this, a lot of the trials, the clinical trials already did. They find use the target on the gut microbiota to find some method to treat the alcohol liver diseases. So you can I found in some Indian in the New Delhi. So some Indian the research team also do the work, use the fecal microbiome transplantation to treat alcoholic diseases. Severe alcoholic hepatitis. So this is also tried. I think more researches we need to do, like we use the antibiotics or the prebiotics, also or probiotics or some fecal microbiome transplantation, and a lot of methods that focus on the bacteria can improve the alcohol liver diseases, I think. And the second, we want to talk about the liver cancer, and we know the gut microbiota is related with cancer. That's the first question. And not only liver cancer, other cancer is related. The first evidence is the Marshall. Marshall is a very famous person. They found the Helicobacter in our stomach, and the interesting story. He in order to improve the causality, they drink the Helicobacter. They also drink, and finally to win the Nobel Prize in 2005. And in the National Cancer Center of the Korea, they used a lot of the human data. They found if you treat the Helicobacter, you can find the significantly low the incidence of the gastric cancer, and also compared to the infection and the non-infection, they found also the significant different of the gastro cancer, the stomach cancer, the incidence is totally significantly reduced. And also another interesting thing is the Fuchsiobacterium. Also, scientists find the social bacterium. Originally they live in the tooth, our tooth, but sometimes they found in the cancers and in the colon cancer patients, they found in the stomach some of the Fuchsiobacterium inside. They found a very interesting things. If the Fuchsiobacterium is low, the survival rate is high. If the Fuchsiobacteria is high, the colon cancer person survival the rate is very low. This is very interesting. They also found a very interesting things. The chemotherapy 5FU, 5FU can the 5FU can significantly reduce the Fuchsiobacteria, and another bacteria can also inactivated the 5FU, the chemotherapy, because the chemotherapy and the bacterial gut microbiota, they are interaction. They are interaction. Also, they found in the inside of the tumor, they also find the Fuchsiobacteria inside. Not only liver, they only in the the bacterial tissues they also found. And also they found that if this recurrence patient, they also was more higher the Fuchsiobacteria inside of this, significantly higher inside. And also not only the tumor, in the paratumor, the beside of the tumor, they also find the Fuchsiobacteria is also inside. So that's the reason in the clinic, these doctors, they use the Fuchsiobacteria to do the prediction. Uh, so another case is the gut microbiota and the immune checkpoint like a PD-L1 and PD-1 or PD-L1 the inhibitor like this. This is immune checkpoint inhibitors. So, but actually in the clinic we found some person is a responder and another person is non-responder. The non-responder, so why we what the reason is not effect? We don't know until now we don't know find actually the reason why this not effective with this immune checkpoint the inhibitor. What the major reason? So one the researchers is very interesting. They get the two person, the two group of person. One group is the responder, one group is non-responder. They get the fecal the stool from the this person separately, and then transplant this gut bacteria to the germ-free mouse, and they found the same effect with the human, the receptor and non-receptor. You can find the receptor person, they reduce the size, the effect with the effect with the drugs, the immune checkpoint inhibitors, but non-receptor also the not a response, not a, give the response. So response, not give the response. It's really interesting. That's the sense maybe the inside of the microbiota maybe some the reason. But what the reason? Why they show this effect? What's the main reason? Another person showed the tumor microenvironment. So previously we just think the microenvironment may be the cancer cells together with the immune cells. But recently more and more people, the scientists found that not only the immune cells and human cells, a lot of some bacteria or the bacteria metabolite also do the work inside. For example, some scientists they use antibiotics or high fiber diet to give the patient to give the patient, and they increased the Akkermansia, the bacteria Akkermansia multifida. This also are one of the good bacteria to increase the to decrease the permeability. And they have some bacteria, they have some metabolite like the CDIMP. This metabolite can significantly increase the monocyte activation and secrete the interferon, so they can stimulate the NKT cells, natural killer cells, and then they help to the dendritic cells to increase the anti-tumor response. So not only the liver, and one research team they checked all of the tumor cells, all of the tumor not tumor cells, the tissues like the breast, the lung, the melanoma, all of the cells. They found a lot of the tissues they have the bacteria inside, or bacteria, or the RNA, means 16 RNA inside, or another the metabolite in the tissues. That may be the bacteria or the components they do some effect in the human tumors also. And also they found that the the bacteria or the metabolite the bacteria not in the in all of the area. They just not a random distribution. That means in the some area they do the effect. We said that is intratumor microbiota. They can, if the immune cells will attack the tumor, that bacteria will stopped the immune system to attack the tumor, and also they can improve the metastasis, the tumor metastasis they have. So we check. So the question is, so through what method, which route and which mechanism they can regulate the cancer, the liver cancer. So as we know, the DCA is chemicals can induce the liver cancer in the animals. We always use this the 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 chemicals to induce the oh no, not DCA, no, DMBA, sorry. So we use this DMBA. But the interesting is, if the animals eat the normal diet, they cannot induce the liver cancer. But if the animals eat the high-fat diet, it is very easily to induce the liver cancer. So the scientists think, oh, why is the reason? Why we eat normal food and not the same inducer, but one is not induced, one is induced? What's maybe some reason? Then they use the antibiotics. If we deleted the bacteria, they happen or not. So they used the antibiotics to treat, and they found if we use antibiotic, they are not coming. They are not the bacteria, the carcinoma will not come in, but if we are not use the induced they're saying maybe the bacteria, maybe some reason. Then they try to search the reason. Finally they find the reason. The bacteria will metabolize the bioacid. That is one of bioacid is the cholic acid. The bacteria can the metabolize the bioacid, and this bioacid will this is second type of bioacid, the deoxycholic acid. They will go back to liver and induce the increased the liver cancer. Actually, they activated the hepatostellocyte, and the final they increased the liver carcinoma. And another is the metastasis. They used a lot of the methods, different the myeloma and the melanoma and the lymphoma, the different type, the cancer, the the metastasis model. They found that if you use the antibiotics, they can significantly reduce that means delete the gut microbiota, and this maybe they reduce the metastasis of the tumor. So, and also the immune route. So what type of the immune route they caused? The immune the route through the immune. What type of the immune route? They detect all of the immune cells. They found like the T cells, CD4, CD8, B cells, NK cells, gamma delta T cells, and both all of the adaptive immune and the innate immune cells. They found the NKT cells is the final reason they found the NKT cells are significantly increased if you use deleted bacteria. So deleted bacteria infect the NKT cell, maybe is one of the reasons. They also check in the human subjects also. They found in the human cells, they found is the CXCL16 is one of the chemokines to recruitment the NKT cells. If you treat with some bioexit in vivo, they will significantly increase these chemokines. That means that you can easily to recruitment more NKT cells. They also find some the biox is a metabolite of the gut microbiota. There's a positive correlation with these chemokines. And if we give the brief explanation, as antibiotics can reduce the gut microbiota and also reduce the second bioacid reduced. That means second reduced and the first were increased, and also increased the CLCX chemokines increased, and increased the NK T cells, and final the liver cancer and metastasis will be reduced. This is one of the reason of the microbiota related with the liver cancer and metastasis. And the third, I want to talk about the non-alcoholic fatty liver. So as we know, in the Asia also, not only in the South America and the Middle East. The third, I think, is Asia. Asia is too many fatty liver patient is in there. We think we don't know the really reason about that. This is environment, but microbiota is also one of the important environmental factors is there. So let's see that if we in the germ-free and the convention environment, if no effect, if we eat the same food, not only the high-fat diet, the only chill diet, the normal diet, if we know the bacteria in the body, so the body weight, body fat will low. That means, and also they showed the picture also showed a lot of fat in the liver, and also liver the TG also increased lipid will increase very fast, and that means that means the bacteria maybe the gut bacteria maybe increase the energy increase and increase the fat accumulation through also we can think that is increased through the energy harvest from the diet, the even low diet. And one interesting case is this is the twins from American, I think, but one the sister, one person is thin, one person is fat. They also increased. They also scientists do the experiment. They also the obese twins transfer the bacteria to the mouse and also to the mouse, and the mouse showed the same effect of the human. That means microbiota can change our metabolite also. Yeah, and also we eat the high-fat diet food. We eat high-fat diet. If we have bacteria, there will more body weight. That means in germ-free conditions, they were low side. That means the gut microbiota can do the energy harvest the work from the food and make the metabolism more fast. And also they do when the animals was young, when he's young, he give the low dose of the antibiotics like penicillin. There maybe the bacteria will be totally changed in the early life. They were changed. Then this mouse can become very fat. They can become very fat. And if we do the same work as the previously human, we've transferred the fat mice gut microbiome to the germ-free mice. They also show the same effect. That means that's the reason gut microbiota is really related with the fat lipid accumulation. So that's the reason why a lot of the farmers, when they give the antibiotics, give to the animals and give to the fishes and they want to make the animals more fat and more heavy. That is not good for our health because these animals also change our microbiota. Is and how can the gut microbiota can cause the fat, cause the liver? That's one I think 10 years ago already one scientist give this famous the theory. That means we know the endotoxin means the lipopolysaccharide. This live in the surface of the gram-negative bacteria, the gram-negative bacteria. If this endotoxin, we eat the high-fat diet, then they will change the bacteria and increase the permeability. Then this endotoxin will absorbed and induce the endotoxemia and become the inflammation. And long time, the low-grade inflammation will increase the metabolic diseases will will lead to result in the metabolic disorders. And also, they checked the metabolism. They checked the CD14. CD14 is a deliver for the it's a deliver role to the LPS, means endotoxin. This deliver for this and through the toll-like receptor 4 and transfer the signaling and to the nuclear and become low grade inflammation. And this low grade inflammation can induce the fatty liver. And also in human, how do we do that? In the fatty liver person, we also find their permeability is about 2 times doubles higher than normal healthy person. So that means in the high fat diet, the NAFLD means fatty liver person, their permeability is really high. And also they found the SIBO means intestine, the small intestine bacterial overgrowth. Also the more than the possibility is more than the healthy person, the fatty diet person. And also we found that the endotoxin, the toll-like receptor 4 expression in the NASH person, so increased. In the fatty liver person will also increased. And then next, I want to see that is the Formibutus bacteria. So we previously we said the best higher is in the human body is the phylum is Formicutus and the Bacteroidetes. In the obese person, they always showed the high Formicutus and the low Bacteroidetes. But if you become the lean person, the reduce your body weight, your Formicutus will decrease and the Bacteroidetes will increase. That may be that means the pattern, the change all of the pattern. And also the NASH and the NASH person, the fatty liver person also show the similar pattern of the obese person, also the showed pattern. That's what we can see if we reduce the reduction, reduce the bacteria death and increase the Formicutus. The Formicutus mostly reduce the butyrate, so that's the reason the energy will increase and the extraction is decreased. So that's the reason lipid will accumulation and induce the fatty liver and to the NASH. And not only for this, and some endogenous ethanol also, the alcohol also produced. That means the bacteria in our some very few of person they can make the alcohol by himself as through but actually is by the bacteria. That means the special bacteria in the fatty liver patients, they are more than normal and obese person. They have the special bacteria, and this bacteria can make alcohol. They have the special enzyme. They can fermentate the food and make the alcohol. And also in the clinic, in the NASH patients, they can find more alcohol than the normal and obese person, NASH. And so they can see the NASH person and the gut microbiota pattern is totally different with normal and obese person also. So not only for this, I just give the example. So a lot of the pathways related with the fatty liver through the gut microbiota. So we can see in the future we how use the bacteria. So what of the strategy for the microbiota? Use the microbiota to find the therapy use medicine. So and I think also there are 2 types of this. One is target. That means the specific bacteria so we can change. Use the technology we can change for the bacteria to that means specifically the modificate the bacteria can improve the diseases. Another is the normal method is on target, means use a lot of method, prebiotics, probiotics, symbiotics, or the postbiotics to also including the FMT, the fecal microbiome transplantation, also can improve the total structure. And both are 2 methods. I think in the future, we will not use only the one therapy to treat all of the diseases. But because of the one therapy, some person effect, some person no effect. We also to think in the future, we as the personalized the enterotype is very important, including the not only bacteria, also the virus, also inside. There's a personalized enterotype. We need to distinguish this and make the different therapy. That is the precision medicine. I think this will be the in the future way for our drug development. Mm. And last one, I will summarize the current effect of the gut microbiome research. Gut microbiome is the leading edge of the scientist research, and it's very important. The basic research and the translation research and government is now is expanding in the world, and so we need to do this, and a lot of the chance in there. And the gut microbiome definitely control to our health and the disease. So. But only I need to want to say the gut microbiome is only one factor, environmental factor for that. So a lot of the causality of the gut microbiome disease should be the research should be studied, and also the gut microbiome research is also still in the early stage because of only bacteria, another virus, archaea, a lot of things not to be researched recently. So. So last one I want to say, I've got to say thank you and thank you for your listening, and so I want to emphasize. Please don't forget this small bacteria, small bugs, and they are always together with you, and we will drink together, we will eat together. Even you eat drugs, they can metabolism all together with you. So don't forget it. And welcome to any questions. Thank you.
Well, thank you, Professor Wang, for the lecture. So I invite questions from the judges.
Yes. Okay.
Hi, Mon. Can I go ahead with the question?
Yes,
Yes.
Dr. Walcott.
Yes, great. Thanks for that. Okay, wow, that was an interesting presentation, Professor Wang. I thoroughly enjoyed it. So, one question that jumped out at me was from your early slide. There's a slide 15. I was really fascinated from the get-go about the different types of the bacteria, the pathogenic and the opportunistic and so on. So I'm wondering, I'm really curious. You mentioned that in 1670, as far back as 1670, the microorganisms were discovered. How were they discovered? You have any idea about that?
Yeah, you mean how to discover the drugs?
No, the actual bacteria themselves.
Ah,
If you go back to your if you go back to your slide 15,
wait,
you had a
wait,
nice
I will
little
check
table
it.
there. Yeah.
15. Yeah. I will
Yeah.
share it. Wait a minute.
Oh, sure. Yeah.
Yeah, I will share it. This one or wait. This one or this one?
No, but yeah, right here. Right. So
Okay.
as far back, no,
Yeah,
the
so
one
the-
before,
yeah, so
that's 15. Yeah, go back by one.
This one.
No, this is this one here, 15. Right. So at the very top, you have that the bacteria were discovered. 14. Right
14.
here. Right
Oh,
on this
okay.
one. Yeah. So you see the date there, 1670. So with their limited technology, how were they able to determine the bacteria and the different types? This is
Differentiate the
number
types.
Yeah, this is number 14 here. So somehow it's not freezing on 15.
Yes, we always use the genomes in the genes, but most of the bacteria they show the different genomes. So we we have the database that show in the scientist we use the common database to distinguish the bacteria uses the genomes. Like the species, we almost the 19 or 50%. we say this is the which type of the bacteria identification this bacteria. So that's many. I saw the many bacteroidetes and formiculus is the best higher. Another is very small, but they only this both two is exist in our human or animal gut, but in the different animals, the pattern is totally different. So you means how to distinguish?
Yeah. Can you slide forward by one, just by one slide? This is number 14. Right here. So number 15. Right. This is the one I was looking at with the 1670 date.
1670.
Yeah, go all the way to the top on the right side of the tape. Right there. Yes, you're correct. Yes, the discovery of how were they able to discover the microorganisms?
Discover the microorganisms.
Yes.
Yes, maybe they find. I'm not know the detailed story. There's the father of the microbiome, Leeuwenhoek. They always see the small things. Maybe they were effective to our bodies. And they found, I forgot the detailed story. Maybe they found sometimes the food will become food or water or something. They will become. They'll become something happen. Something that means proliferation. Something proliferation, but they don't know what is this. If we put the water in the outside or something happened in the water. Previously, maybe they think there's some seed inside or some other. They don't know what it is. And these scientists, they make this microscope. I showed it here. They can enlarge the small things to 266 times, and then he found this bacteria.
Yes.
First. Not only find the bacteria, they also find the cells also.
Yeah, this was really interesting. And then, if I may, as I'm on the floor, can I ask one more question?
Yes.
About on slide 26 though, I was wondering if it is you had any information about different alcoholic drinks across in my country here, where we're getting into the Christmas season. Persons are beginning to buy up alcohol, and just recently our government raised the prices on alcoholic beverages. So did you come across any information about the alcohol in different drinks and the impact on the what you have here? Yeah, is it just specifically alcohol or different drinks?
Yeah, so you mean the alcohol drinking different, maybe different for the impact to the gut microbiota?
Yes, like whiskey versus rum versus beer.
This is totally different. I think it depends on not on the alcohol, because in the whiskey or the wine and beer, this not only in the alcohol, they also have another things, because in wine we know a lot of the some antioxidant component inside.
Correct.
Inside. That's maybe some show the good effect, not only the alcohol. This is only experiment for the animals, even in the human. They also just think more. That's means high dose, I think. But low dose is not the effect. I don't know this how much. Not sure for that. This is the 3.5 gram per kg. Well, I don't know this is high or low dose for that. But actually I think low dose is not like this. Will significantly increase the endotoxin, I think. But this need to do the experiment. For the low dose of the alcohol, they can induce the low grade inflammation to our body. That is an interesting question. We can try. So even low dose. But I think the alcohol can make us happy. But also, in my thinking, the wine is more good than the whiskey.
Yes, for the antioxidants and so on. Yes, okay, great. Yes, thanks so much for your presentation. So I think that's it for me, for my questions.
Thank you, Professor Wang. for the knowledgeable lecture and your time. And I again congratulate for the APP award, and the respective certification will be released today evening, and I will send to you. So thanks a lot. I will feel happy to involve you in future APP conferences. Thank you.
Thank you very much.
Yeah. So we have our next speaker, Professor. Let me go to schedule. Professor Gajanan Kodake, who is also from Dongguk University, Seoul, Republic of Korea, and who is presently holding the secretary post of the APP Korean International Branch. So without taking not much time, I invite Dr. Ghodake to deliver his speech. Dr. Ghodake, please.
Yeah, thank you. I'm audible?
Yes,
Hello?
you are audible. You can share your
Okay.
slides.
Okay, great. Just a minute. Hello? Shall I start?
Yes, you can go ahead.
Thank you, all of you, for a nice introduction. I greatly appreciate esteemed Professor Raju Dayaji for his long association with us and our colleagues. So I appreciate his friendly nature, approaching people, motivating, and bringing those international talents for our students. I would like to talk today about the sustainable agriculture, climate resilience issues right now we are facing across the globe. We know that it's not just a minute, there is some kind of echo here. Hello? Let me control the...
Yes.
There was echo. There is still echo. Can you correct that?
Yes, organizer.
Hey,
There is echo in the system.
Yeah. I'm getting my voice back. So just correct their speaker.
Hello.
Yes. I'm getting my voice back.
Okay, I'm calling.
Just check the speaker if there is some issue. Hello? I think I let me get out from the one screen.
Yeah, Dr. Godake, the topic was a little bit different what you provided.
Actually, I was not prepared that particularly that. So whatever ready, I'm going to talk about. That's not an issue because it's also one of the topic of my interest.
Okay.
And it will be also. Let me leave the session from there. One session I will continue from here. Okay. Now no issue. You mean which one I propose analytical? This one?
1 minute.
Can you check? I have both presentations. I can go either this analytical or climate resilient agriculture. Which one you prefer? That's fine.
1 minute, 1 minute. I'm looking. Your topic was translational nanomedicine for antibacterial and anti-cancer therapies.
I think we are not able to talk on that because I get
Okay.
out of the mind about our today's. So
Okay, no problem.
let's
You
go
can
either one of
go
them.
for how.
And I would love
Okay.
to talk because recently I talk about analytical science diagnostic. So and also about the agriculture, but agriculture will be the much interesting for all of us.
Okay.
Yeah, sure. So today we will talk about how we can develop the future agriculture, about the sustainability and addressing the issues of the farmers, where the global sustainability and circular economy is being not it been established, because it can transform the food system as well as the economy of the especially on the biocircular economy. So
You
that
can share
concern.
the slides.
Yeah, sure. Just a minute. I missed to share the slide.
Yeah, you can go ahead.
Oh, I see. There is some concern. So one more thing, I will switch to the web. Okay, that's fine.
It's okay. If you are taking time, then we can ask
Because
the next speaker.
the presentation is on the web, and just are you recording my speech, right?
Yes,
Just a minute.
recording is going
Okay.
on.
Then I will not. I will just started my phone as well. So I will just quickly move to the web. Here is the link. Is now fine?
Yeah, it's fine.
Yeah, it's fine, right? Okay, perfect. So sorry for inconvenience. I am Professor Gajanan Godke. I am working as a associate professor in Dangook University. And first of all, I congratulate esteemed Professor Raju Deyaji for his unwavering support and his effort to bring this our society and this project alive, and to connect the esteemed scholars across the globe to serve our students and to have the motivation and inspiration for them. So let me continue the introduction about the concern about the farming. So far, from the ancient time when the farming started, it was based on the natural ways, and then it was continued until the last century. But what happened after the Second World War? There was different chemical industries emerged, and those after the World War, the chemical industries turned into the agrochemicals. And those agrochemicals we called as green revolution, and somehow we brought the food security. But the now concern is the greater, where we can see the lot of issues arise from the food security with uh, on the way that we have to suffering either like biodiversity losses, human health concerns. Once upon a time, the ancient philosopher said that what upset people is not think themselves, but their judgment about things. So harmony in the nature brings the abundance, but so far we see that our Vedic wisdom is also aligned with that. Harmony in the nature bring abundance. So in the future, we cannot continue with agrochemical like the future. If with that, already you can imagine the day we are spraying. Some farmers are spraying a couple of times a day, and those chemicals are non-target or killing the non-target organisms as well. So we must consider that how we can mitigate the biodiversity loss with the green solutions, and also address the concern about the climate the change. And because of the climate change, the farming practices has been disrupted significantly. So to see the grand global challenge, why we should transform now, because either we don't transform now, we will lose everything: soil, water, and air together. And you know that, and after the industrial revolution, we are producing a lot of air pollutants, as well as causing the water bodies, like for example the industrial waste being some developed countries not being sufficiently treated, and they're directly discharged and they're causing. So climate extreme disrupting the yields as well, because of if the rain can damage whole crops across the region, and then it becomes the great urgent emergent food crisis. So increasing severe droughts is another concern. Some countries devastating floods is there, and those are showing the prolonged ways. Like for example, this year India has experienced extensive rainfall, like from early started from May to then some states like until the September. So this was like long, and the rain get covered, and also in addition to that excessive rain together without the sunlight, it caused a lot of root rots and completely wiped out those existing crops. And suppose there are some regions are drought prone, but they are now experiencing excessive rainfalls. So it's like unpredictable, and I have the intelligence that it being unpredictable from last 3-4 years, and farmers are in distress. So look that if this condition continue, the crop productivity and farmers' livelihood is at great concern. Another is soil degradation and water stress. Suppose there are intensive farming has depleted, like monocropping and all those things matters a lot, because of the millions of hectares being overfed or over irrigated, or somewhere like nutrient runoff because of the excessive rain create to the ocean zone, and groundwater depletion, as well as irrigation dependency regions that becomes serious concerns. Biodiversity collapse, as I already discussed, that biodiversity habitat is under the great concern because of the lot of pollutants, and the farmers are spraying. Some of the farmers are spraying 2 times, 3 times of those sprays, and they're causing also death of the non-target organisms, including some insects and honeybees. So this is also great concern of ecosystem functioning. Agriculture productivity is also rely on those biodiversity around the farms. So if there is no biodiversity around the farms, suppose insects and birds together, that will be the failure. And in the future, that will be alone, the pesticides and agrochemicals will alone responsible for the greater food security in the future. Food security and equity, we should consider that food security is not only come from the agrochemicals. It will be also the reason that the food security will be also great crisis because of the agrochemicals as well, as I described. Because once the soil microbes are lost, soil fertility will be lost, and then this infertile land will be non-productive, and farmer will be again in distress. in the future. Despite producing enough calories globally, inequitable access of nutrition and food persists rising cost. Plus, it will be again a lot of implications if there is, because it's healthy diet beyond the reach of the billion peoples, and those things, if happens, it will be like vicious cycle. It will continue affect the series of the other concerns. In this century, it's very important to us. We must look into the produce the more food without damaging the environment. That should be our prior goal, because environment footprints are, as I told you, that they are creating a number of issues. Like it's also required that that's why we require the innovation as the responsible researchers and scientists. We should have the collaborations and look into the fundamental systems and redesign those future systems where we can look. So let's see how productivity and input and adaptation and mitigation we can bring in the future. What do you mean by climate resilient agriculture really means for all of us? That's what, let me give the more clarification about this. Climate resilient agriculture represent the holistic transformation of the food systems. So it can be also address the adaptation, mitigation, productivity, social equity together, and social equity together. We can have the it is not just like producing weatherproofing farms, like like protected farm and greenhouses. It's about redesigning entire agriculture ecosystems, especially for the long-term sustainability. So the mitigation together with the system view, farm to market integration, and stable yield and the fair livelihood of the farmers, and adaptation should stand to the shocks of the climate variabilities. So this transformation is like very urgent like situation. We should look into the food system pressures, identifying the root causes. So the root causes are like monoculture dependency, because you know that even though there we have the different policies to diversify the agriculture systems, but monoculture dependency, the monoculture is also another culprit where it can create the vulnerability to pest, disease, the market kind of shocks, while depleting soil biodiversity as well, because it also ruins the soil because of the monoculture and repetitive practices of the same crop varieties and synthetic inputs to generate more productivity. Another issue is like linear material flow. What does that mean? That make the waste. For example, we have to develop the model that will dominate agriculture, where the input become the waste rather than the recycling back, because environment issues like remain unpriced and creating a lot of the complications. That's why we have to see the linear, like it's like recycling, like some kind of waste again, and it creates no economy either. So that's why linear material flow, and it makes sure that, for example, the input of the fertilizers gone through the roots and it converted to the food rather than the runoff and the waste. So another issue is like discontinued supply chains and some kind of the poor practices in the storage facilities, also causing the losses of the post-harvest yields and the products. Information is not symmetry there. There are also issues still. We have to look into the how we can deliver the right information to the farmers, capturing only not the small fraction of the final product value. That means that when we look into the controlling every aspect of the food security together and data individually, like poor monitoring system, right? Still we have the suffered the poor weak extension system, limited access to the finance, prevent the farmers from the making their informed decisions and access to the resources for improvement. So still there are a lot of scopes. So these system failures are also will be the transformed solutions to address the circular material flow, diversified cropping system, like multi-crop and like companion crop, those kind of like companion crops, in case of that one crop support to another crop together grow. And then there is one popular crop system is called three sisters. Three sisters, like for example, corn is there to support the beans, and for the surface we can grow like some kind of the like pumpkins or some other. So it's like we can utilize like vertical as well as support, and then that kind of the companion we can develop or adapt those kind of practices in the future. Let's see the what kind of the climate smart agriculture practices, how we can build on that. For example, we should protect the soil health to reduce the to reduce the the risk of the future food food security risk. So we can use like reduced tillage, crop cover system, and then some time of the biochar, like just compost applications that will be built the strong, robust soil where we can sequester carbon as well while boosting the fertility. So this is like a holistic approach to the soil health. Instead of just putting the agrochemicals and agro fertilizers, relying on and for certain years it will be like look at, oh, it's like extra yield, but the when the yield will start reduction, the controlling of that yield will be the almost impossible. So diversification, as I told you, monocropping is not the solution, but still it is being largely practiced because of the the different reasons, farmers reasons, and then the water accessibility, and those people change their mind and rely on the one crop. I will not mention those because that may hurt the farmers' sentiments. So intercropping, crop rotation, polyculture, reduce like pest pressure because of this distribution of the pest, and improve soil health because of this companion, and some are like nitrogen fixers, and some are climbers, and some are supporter, and some are like covering the soil surfaces. And this kind of spread, it also reduce the risk across the uh, different aspect, and it's also generate multiple income. For example, small landholder farmers, if he has the multiple products, and he can go and offer 20 products together on the same plant, 2 or 3 hours he can go solar and get out of his house back and also continue. This model can be also good for the small holding as well as the large landholders, and can be biodiversity. Also, we can protect our the ancient seeds, and we can build like foundation of the resilience. Water situation, like micro irrigation is now widely getting adopted. Rainwater harvesting is also being promoted by the governments, and the soil moisture monitoring systems also we can integrate with the AI monitoring data because of the large number of the point the where we can monitor and use the AI for the monitor the moisture conditions, and protecting the watersheds and from the pollution and depletion. As you said that those irresponsible industrialists still pouring their wastewater to the fresh water bodies and the rivers. So this still should be controlled under the stringent environmental laws. And another idea is like fourth pillar, it can be the precision management. For example, applying the right inputs, right dose, and the right time, the right places, minimize the waste, and also the reduce the cost, prevent environment contamination through the data-driven decision making. We can utilize here the AI systems in case of the for the precision management, because precision agriculture is not possible without the data management. These four pillars, if work synergistically and precision management, we can optimize water, soil interventions, and those kind of diversified practices can be very effective together. So it can create the stable yield, stability in the income source and input efficacy as well, like atomic economy. Atomic economy means suppose we put the n number of the input and it convert to the maximum percentage of the input to the final. So climate agriculture is not far distance dream. So we have to just integrate. And another idea is like how we can integrate the agroforestry for the climate resilient agriculture, because agroforestry is very compatible for the climate resilient agriculture strategies. Because of biodiversity is lost, you can see that it's immeasurable, and we cannot even control the losses because of its every day is our soil, water, air is getting damaged, and it will get worsened day by day. So biodiversity is being under serious concern, and the human as a human we have to be also take serious concerns and attention to the how we can reduce the biodiversity losses mitigation, because it's like uncontrolled situation. Like for example, biodiversity, we can not only consider the tree plantation, but also we can ensure that all sort of the biodiversity, pollinators, insects, birds are also get their spaces there. And for example, if we provide the corridor for wildlife movements and soil organic diversity together, it can only the save our uh, ecosystem. Otherwise, as Albert Einstein said, if the honeybee gone from here, there's no life for humans. Imagine one entity in the ecosystem can how much create the damage. Microclimate regulation, for example, the strategic tree plantation may provide different kind of shades for different and wind breaks, temperature moderation, and the wild reduction of the soil as well, and improving the water filtration during the extreme weather. So microclimate regulation, another is like natural insurance policy also. For example, agroforestry is there, and we can control the climate insurance buffering, some kind of maintaining the productivity, as well as ensuring that those productivity during the extreme temperature through the microclimate modulation, because there are different issues in suppose there are high temperature situation, we can plan strategically that how we can create the microclimate to that may be suitable for the another cash crop, and this kind of economic case can strengthen over time and defend the tree mature and provide the valuable products from that tree as well, and continuously improving the underlying soil fertility together. So this system, like for example, there are like spice farms. The spice farms, they are like the multi-layer farming. So they are companion together. And also we can diversify the income. These days, farmers are rely on the one kind of income or maybe two kind of income maximum. So if something wrong happens, they lose everything and they get the distress and they get the vicious cycle of the loan and repay and loan repay, and then it become very issues. Like if the farmers distribute land to the different purposes, some kind of the fooder, some kind of the fruit tree, vegetables together with timber trees, it's like this all different, and some they can add the farm allied businesses as well, small businesses together. Maybe at least I believe that one farmer should have at least 12 kind of income strips. Then farmers can have the comfortable life. So I humbly request that in the future, when the farmers get this kind of idea, try to add one allied business after allied another, because of the, like, even Warren Buffett used to say that keep your eggs in different baskets. So these are the different baskets for the farmers. They should create the baskets. And this carbon sequestration is also one of the agroforestry that can add the climate change mitigation. So how we can relate circular bioeconomy for the rural prosperity? There are farm residues, local processing, energy and enriched soil, compost, biogas are different small projects. So this should be like ubiquitous to the across the nation, and every small village have this kind of together, like it's like integrated system, waste to the value transformation they can convert, and the local economy, I would say local economy, and the reliance on the local the farm producers, as well as the rely on the village products. Establish village level processing units to reduce the post harvest loss, and they can value addition and then get the right values in the market. Also, they can increase the final product value that remains that among the remains with the farming community itself. Their own, they should add the values. So For example, that if they have the waste, they can transform it to the multiple byproducts, and they utilize their among their peer farmers, and then they rely on each other and not rely on the external the agrochemical suppliers. Transforming agriculture waste into valued products will create definitely income opportunities while closing nutrient loops and reducing environment pollution. Also, economy of the village will be the strengthened and become the people lives in the villages and small towns rely on the agriculture, and the agricultural allied businesses will feel the have the goal of the prosperity, because regenerative cycles can benefit farmers, communities, and ecosystem. There are so many stakeholders. In case of the milk production, animal husbandry, there are how many stakeholders? N number of stakeholders are there. So it get directly, indirectly benefited. So there are some nature positive material economy measurement of the impact of the matrices. We will be not able to cover. So let's move to the another. Green chemistry transforms agriculture input. Right now, I will jump to the green chemistry. We talk about a lot like Second World War, and then the chemical industry turned into the agrochemicals, and they convinced and even there at that time situation was like agriculture productivity was the great concern. So they adapted worldwide, and even they named as Green Revolution, and we successfully year by year, decade by decade, we increase the productivity. But we somehow disregarded to the unutilized land or under irrigation lands to come and then increase the productivity instead of the based on the chemical alternative. So I would say that redesigning fertilizers, pesticides as like green chemistry using the nano pesticides, nano fertilizers together, and those green chemistry principles can reduce the toxicity. That should be our priority goals. Maintaining the or improving the agriculture productivity. This transformation can produce different kind of the protect different kind of the health causing agent, like with cancer causing agents, and also preserve the ecosystem, maybe create some safe working conditions for farmers. And so right now you know that high application doses and significant losses, runoff and contamination of water system is also creating the multi-drug resistance issue. Multi-drug resistance is also we farmers spray the antibiotics, and they use antibiotics in animal husbandry. They finally reach to the water bodies, and that also creating like emergent issue like multi-drug resistance. So controlled release, slow release, for example, formulation, this kind of new formulation, reducing the waste, biodegradable carriers, preventing accumulation, and all those kind of things we can also consider in our research, where we can address concern like. And biotechnology and smart material, as I told that biotechnology can play the like nanofertilizer, pesticides, seed enhancement technologies, real-time sensors, and safety kind of development, like those kind of will be safe for the consumption as well. So nanofertilizers, is there hope? Of course, there is hope for the nanofertilizers. For example, nano urea is getting much popular, and nano urea is the cost effective and also not causing that much environment pollutions. Balancing innovation with precautions, because innovation and with the concerns and the implications is not acceptable. We should see the long-term environment persistence, soil organic beneficial insect impact assessment, and overall ecosystem, human health exposure pathway analysis. It's just like we should be like holistic analysis, and then after we can scalable green synthesis or safer synthesis without toxic precursors or less toxic precursors at least. So regular standards we should implement, and technological advancement slowly, because we can not switch the chemical agriculture to the organic agriculture that what we can experience in the Sri Lanka, that can be fatal for economy. And so digital agriculture and artificial intelligence, this is also like new thing. We can also AI made decision, market integration, design for inclusion, these kind of things. For example, design agriculture must be prioritize usability, offering like interface for local languages. For example, some information is only in English, then local farmers, how to rely on the translation, and those kind of things can be like mistranslated and misused for that. Affordability also matters a lot, because if we design something and it's expensive, because you know that I came to know that many farmers are suffering from the expensive fertilizers and pesticides we import from the multinational companies. Affordability is also one of the important factor we can look into the expensive equipments. There are some, like for example tractor. If there is one tractor, the equipment together it goes like minimum 15 lakhs, 17 lakhs rupees. The farmers cannot afford that much. And how to then equipment upgradation. And there is another issue, like because of the generation after generation, the land fragmentation, the small landholders cannot afford the machineries. So successful platforms we can combine together, and remote sensing, traditional knowledge. Also, we have to look into the validating farmers' observation. Sometimes we should collect the data what the farmers are observing, and then respecting their local expertise as well. Some farmers are quite genius and intelligence. Those can be also. We can also look into the cooperative shares, share some kind of infrastructure, reducing individual cost while building the collective capacity. The farmers should also come into the together. Another, how you will say that what are you talking about, the machine learning and AI? Farmers have small landholders, they are suffering, they are trade stress. How the AI can help them? AI is not the farmers are going by, but the agencies can provide the analytical data using the AI, and that should be like accessible. Look, machine learning algorithms can analyze the all sort of the patterns across 1,000 prompts, 1,000 forecast pest control, then diagnostic, preventive measures, optimize like planting windows, recommends like precise nutrient application for the local. So we can reduce the cost of the farmers. Farmer dashboard is another idea where we can look into the intuitive interface, local languages translation, as I told you earlier, and IoT sensing networks, soil moisture stations, weather sensors, and then satellite image, kind of providing continuous environmental monitoring, and that is should be accurate somehow. People should believe, and farmers should trust. Oh, that must be true. Not must be true, but it's a true. Like so that should be like those kind of the guesswork should be not there. Oh, people are confused and have the unpredictable and uncertainty and this kind of no more. So we should be looking to the data driven uh, monitoring and irrigation and input scheduling. Like input, like for example, fertilizers input. There should be some schedule or some, if there is required, then should be. Otherwise, it's not should be blind. So that kind of things should be coming to control. Sustainable diet and demands like transformation. For example, supply side innovation, like we should construct some pattern, like we can promote nutrient post harvest and based on the plant forward diets, reduce the pressure on the land. Because if we consume the animal the products, you know that how much kg of the food we have to feed to make that animal 100 kg. So that maybe at least 50 times of the consumption. That means if we are consuming one animal and uh, then that requires 50 times of the grains, that means how much of food security issue we can cause if we have based on animal diet. So the plant-based diet and also water resources while improving the public health. Right now you can see the water resources has been totally polluted. Imagine if in the childhood you will be very physically and very safely, and without any doubt you must be enjoyed the water from the water streams in the rainy seasons. So it's not the concern, like it's planetary health together, nutrition of nutritious, affordability, sustainability, and it's like a optimization of the diets, nutrition parts, and affordability of the nutrition. Because of the malnutrition, we can see that how what kind of implications we are having. So I would also look into the stoic some principles in agriculture where we can produce because of the farmers' distress. We should look into this kind of stoic principles where like farmers are resilient. I know, but still they have some limits. Because of the crisis after crisis and distress, they lose their resilience and their patience, and that's why we should always protect our farmers through the education, public education. Resilience in terms of we can have the prepare and diversify risk and bring the more resilient power to them, and optimize timing those we can make the farmer discipline, so it can also reduce the cost. In case of the courage also, we can look into the courage, resilience together with temperance, we can conserve the water and energy. So also the courage in terms of the trial, innovation, and the measure. Farmers should not do that. We have the national institutions for the farmer and agriculture research. So they should be very close connected. They should not sit into their laboratories and do research and some sort of the publication, but they should look into the what kind of technologies are available across the globe, and they also engage into the uh, translating technologies to the ground. So in case of the ancient Stoic philosophy, it's called aporox, surprising relevant guidance for the modern agriculture as well as our personal level. Because that or this core value like wisdom, courage, justice together, and temperance, and some kind of the translate it to directly farming practices that will build the resilient and optimize resource used, and we can foster the long-term thinking and sustainability of the farmer. In addition to the Stoic, what Vedic we are just like disregarding, because in case of the chemical-based agriculture, Vedic ecological wisdom for the modern agriculture, how we can slowly turn into those kind of principles. For example, Rita means cosmic order, like aligning agriculture operate with seasonal and lunar cycle with respect to the natural rhythm, optimizing the planting, some kind of planting time, harvesting, and some harvesting times and resource availability. So this cosmic order, right now farmers are farming like without looking into the crop cycle or the weather conditions. So that can be like, because they look into the market price based, so they are not maybe get risk of the environment climate change concern because of the change in the climate. It is not maybe suitable for those crop where they are targeting. Soil as a living mother, because in Vedic, soil is called Dharani Mata. We call Dharani Mata, right? So we should have that respect. If necessary, we should put into chemicals to protect, and this should be like target specific and not like spraying just like everywhere covering the soil and then not ignore. So this is happening. We should bring not only saying just Dharani Mata, no, you should have that respect in heart and protect and have that kind of new tools to only spray on the target areas. Like we should look into the tree plants out where the leaves where why we should spray. Like when spraying, they just like get wet, the soil get wet. So that means we are responsibly doing that. So some Panchagavya traditions also we can look into the for example traditional organic cow-based products, some extract fermentation, like create nutrient rich liquids, and that can avoid chemicals. And cultural continuity, because of the maintaining agriculture tradition, social capital, environment together. And imagine even the poor person, if we offer two options like the residue free and with residue, they will prefer the residue free. We cannot say there is no demand, there is full demand, but there we should develop the system, and even the buyers should also openly come up and demand the residue free agriculture products, so farmers get motivation for the looking into the back and putting the emphasis and cleaning the soil residues and then producing. The another is like traditional knowledge, like it's called like they are considered as superstition. If that's aligned with nature principles, why we should call it superstition? This is the like a kind of misconception being created, like, oh, if somebody is going to be back to the natural and traditional way, they will be like regarded as like either superstition or the practice of the course. So this should be changed. We should honor our cultural wisdom. We should apply the contemporary measurements as well as our original long back the system, like 2 decades before, or every village of the India was self-reliant. They were having 12 kind of the artist and the support, like even the metallurgist and the pottery maker, shoemaker, everybody, and including the other agrosystem and the microbial diversity that was intact. And but that time, if we implement only for example, biopesticides, there are some biocontrol agents as well, like biocontrol pathogen as well. So those has been still ignored. We should have special policies and to take into the serious consideration. See here, Vedic philosophy together with Stoic principles and modern science together. It's not like I would say just only go Vedic. Some people are doing that, right? Or some people may say natural. No, we should combine together. For example, like in case of the Vedic, we should just see the science and the Vedic principles together, ethics and the Vedics together, regional agro-system. And in case of the Stoic, we should see the science, ethics together, and modern agriculture. The science should be emphasized. Science align with the Vedic and Stoic principles together, and then combine a new system for the new agriculture. Policy, finance, and subsidies, and these are already going. Maybe risk management tools are not being yet implemented. Blended finance vehicles will be needed because farmers are still relying on the private lenders. And the policy, I would not talk about the policy because of this is not our concern. Let's move into the community capital, social infrastructure. Yeah, this may be quite important because of our community still should have the combined efforts, not alone separatism. So because many data are common together and their interests and their passions, they are together, like they should find their unions and groups together and so address their issues with joining hands together. So this will be the not concerned about like how we can transform the agriculture roadmap for 2030, 2040, and 2050. So this is the maybe specifically it may be for the policy decisions. Yes, sustainable development goals and key performance indicators. For example, scientific foundation, ethical framework, ecological alignment together with yield, risk assessment, net greenhouse gas reduction, and biodiversity protection together with carbon neutrality or negativity target, reduce vulnerability to the climate shocks. So this together, maybe we can achieve. If we follow those kind of ways, we can adopt. We can develop new instruments, invest little bit, like direct resources toward the people and platforms, to training programs, building the farmer capacity, digital infrastructure together, co-create. I would say like join initiate collaborative pilot projects together and scientist industry partnerships. So those kind of engagement should be there. Science, philosophy, plus community together, we can have the climate resilience. It's not one person job or one organization job. It's together. We have to join hands together, and the collective efforts, collective evolution together. Ethical philosophy can guide this kind of the future decisions, and the community activity implementation can bring the change. Like these elements working together can enable durable transformation, not for the like decade or 2 decade, because its world is changing like a bullet speed dynamic thing. If we bring some system that will be sustainable for durable, longer transformation. So that will be much appreciated. Thank you so much for the listening. And if I will be very delighted to listen your questions. Thank you so much.
Thank you, Professor, for guiding us on agriculture aspects and how we can protect the earth, especially in terms of agriculture point of view. We by simply saying mother, that point was heart touching. It is not enough. We have to protect the earth in many ways. So, if any question is there from the judges or any audience, any question? Okay, in absence of any question, so I thank Professor Kodake for your time as well as your support toward the Korean International Branch. Hope the branch will move up to good heights in your able leadership with Professor Wang. I remember in March when I visited Dongguk University and you introduced me to Professor Wang and we visited your and his lab. It was very nice experience by seeing our scientific bodies working in different parts of world and so efficiently they are publishing the research. So thank you very much for your support. So we will be in touch.
I would like to request all of them to turn on their camera. I would like to see some personal introduction. Balaji, Dindaji. Yeah, principal sir. Yeah, come, let's have the introduction, some kind of small, yeah. Balaji, Dr. Balaji.
Professor Balaji, you are here?
Okay. Okay.
Professor Das?
Yeah. I think I request all of you just turn on your cameras. All shy guys. Okay, no issue.
Okay, we'll catch them at some other point.
Yeah.
Thank
No
you,
issue, no
thank
issue.
you, Professor,
No, it's a great pleasure.
for your time, and we will be in touch in March.
Sure.
You have another event with us.
Sure.
Okay, thank you. Yes, our next speaker is none other than Professor Neeraj Kumar Filoria, who is our president of APP Malaysian International Branch and presently working as professor in capacity of professor of pharmaceutical and medicinal chemistry at AIMST University, Malaysia. Professor Floria, you can start your presentation.
Thank you, Dr. Daya. Thank you so much. So I'll share my screen. I hope my screen is visible.
Yes, it is.
Yeah, thank you.
But you can go on presentation mode.
Yes. So before I start my presentation, let me thank to the APP and the organizers for organizing such a wonderful platform wherein we can share our views. Thank you so much to all.
Professor,
today,
can you put your video on for some time?
Okay, yeah, I can.
So then you can, we are clicking some photos in between. So
Okay.
if your face.
No worries. Okay, thank you. So once again, thank you to the organizing team, especially the APP, for giving me a platform to share my views over. I'm going to explain something It's very old. We can say it as a old wine in new bottle. That is healing with the light and orthomolecules, a scientific insight. So you can, with the topic itself, you can make out like I'm going to talk about light therapy and orthomolecules close to the nature. So already we had a discussion over the soil and its all the aspects related to it. Now it is something close to it. We'll be talking on the nature only. So firstly, we'll talk about the first talk which I'll give on a light, or we call it as a phototherapy, or we call it as a chromotherapy also. Um, we all know the light is our eternal source for life, and the colors brings well-being to all of us. And this is the light is a radiance that vibrates within our body and dispels the disease ultimately. So before going to the light therapy, let me introduce where this light therapy has been categorized. As per the NIH, we have there the NCCIH now, National Center of Complementary and Integrative Health. Earlier, it used to be called as a National Center of Complementary and Alternative Medicine. So we have the five domains there. One is the alternative medical system, where we have the Ayurveda, Siddha, Unani, all that. Then we have the mind-body interventions. We have the biological-based therapies. We got manipulative and body-based method. And also we have the fifth domain is energy therapy. So this light therapy or phototherapy or chromotherapy comes under the domain of energy therapy. Now to introduce about, you can see this beautiful image here. So we all know that our nature is filled with colors, and these colors serve as energy which influences all of us, isn't it? So in a sunlight also, we know the colors have got different impacts on our plants, animals, or human species. So each color affects our human body uniquely with its intensity. So we know the physics behind any object will exhibit color when they absorb or reflect certain wavelength of light and stimulate our cone cells in the eye, then we can see the color. So this phototherapy, also called as chromotherapy, also called as light therapy, in this treatment, what happens basically, we apply some electromagnetic waves, normally the visible one, which ranges from 380 to 780 nanometer, we all know about. So normally these radiations we are applying for the treatment of various disease while promoting the overall health and well-being of all of us. So, to talk about the history of light therapy, actually, it's very dated back since the Ayurveda time. We had this light therapy treatment at that time also, but we believe in evidences and documents. So, this light therapy actually it is since Ayurveda and the Siddha, all that time people were using it. Then at that time they used to use the light and the color halls. You see, still you can see the old the tombs and all where the people used to have the jewels. From there, the light used to pass. Even we use some stones nowadays, right, for the light. So 2,500 years before itself, in the documented what is, the Pythagoras first time they applied the light therapeutically. And in current scenario, Neil Fiensen is the person who is called as the pioneer of modern chromotherapy. He has studied how we can utilize the light for the treatment of healing the wounds. For this, he received the Nobel Prize also, basically. Especially, he got this Nobel Prize for this lupus vulgaris with a concentrated light radiation. He did the treatment, that's UV blue light, we say. With that, he made the treatment. So then with that, he got the Nobel Prize. I'll come on it. You must be thinking, why? What is the implication of this light therapy for all of us? Yes, because nowadays, all of us want some novel research, some novel idea. Maybe by this, what I'm sharing with you today, probably you'll get some new idea. Like in 1933 itself, the Dinshaw Gadiali, he was an Indian only, but in New Jersey, he was, been reported, like he explained the colored rays has got a therapeutic effect. And in his experiment, he exposed colitis disease for the light from lamp fitted with the indigo color glass filter, and he could treat it. So, what is the implication where, in which condition we can apply this phototherapy? We can apply in various conditions like in stress, depression, aggression, anxiety, high blood pressure, sleep disorder, and many cancers, skin infection, and jaundice, etc. You know, you see, psychologically also light has got a lot of effect on our body. Example, you see in the Western countries where the light comes only for the 6 months. All time the sunset is there. I mean, you can't see the sun. It's all time the ice age. So there are a lot of depression cases. You see, in the winter season also in India, normally what happens? We go for the light exposure. So that's why this light is been given for a treatment for variety of condition. We'll come on it later. One biggest example now today, almost in every house you will hear like the newborn baby got a jaundice called hyperbilirubinemia. So it is the most common use of phototherapy wherein we use. Here normally we use the blue light, which break down your excess bilirubin in the child and a baby blood, that causes the skin and eyes to be yellow and thereby prevents the brain damage called as connectaris. So here it's the biggest example we all are using in our daily life since a new baby is born. That time also we use. So one study was there in Iran. It was being done. It showed that the phototherapy can reduce the serum level of 2,5 hydroxy vitamin D in 60 icteric neonate. They did a clinical trial and who were having this hyperbilirubinemia. So with them, they exposed to the this radiation and they could, lower your this. They could treat this hyperbilirubinemia. So thereby this light radiation normally we use in various conditions like psoriasis, eczema, dermatitis, acne, scars. Like for psoriasis, we say there is no treatment, but yes, with the radiation, there's a treatment for this. So what does this phototherapy does? It reduces your inflammation, minimize the itchiness, prevent the flare-ups and chronic skin issues. You see, one biggest example I'll tell you for this light therapy, we do our treatment daily and we never know. For example, let's say you are running around the hot sun on a beach. You are on a hot sun beach and the radiation are very high. You just measure your blood pressure by that time. You'll find your blood pressure will be high. So same time, now immediately we'll make you wear the black goggles or green goggles. So then we can measure your blood pressure. It will reduce. It will come to the normal. So that means the light therapy works. So blood pressure controlling means ultimately it can be used for your treatment. So it is the biggest example I'm telling that we use commonly. You are traveling with a car. So you can find out the same time, like, yes, you are getting treated. So there are certain techniques with which you can apply this phototherapy. One is by looking at particular color to desired response in your body. I mean, one is by, through the eyes. And secondly is you can do the direct reflection on your body parts, the color. Actually, as I told, it's an Asian science. If you see, majority in the India, they used to wear a lot of stones in their, the fingers, correct? So they feel when the light radiation will pass through, it will have an effect on our body. Sometimes we say the different stones are there, different crystals are there. That has got a different effect. Like we say the white stone, it has got a cooling effect. We say the red stone, it is very hot. Your blood pressure will rise. So it will give a different effect. So this color therapist or you say the light phototherapists, they believe that these light or colors enters our body through eyes or skin. So each color has got different frequency, and different frequency means they will elicit different effects. Normally, if you want to have a stimulatory effect in your body, you need to have a warm color for stimulation, whereas the cool color for calming effect. Basically, we all know, and there are three primary colors. One is red, other is blue, then yellow. A mix of this prepares the secondary color. That's red and blue gives your violet, red and yellow produces your orange, blue and yellow makes green. So likewise, we have the different secondary colors. And mixing this primary with the secondary colors creates different tertiary colors. Correct. So like example, magenta color consists of 2/3 of the red and 1/3 of the blue. Likewise, we have the different colors. Okay. So coming to your principle, how does, it gives an effect in our body. So we all know like our body physiology depends on light, like example, circadian rhythm or sleep-wake cycle. It all depends on the light. So when this physiology is disturbed, or this biological clock is, or circadian rhythm or sleep wake cycle is disturbed, we get numerous diseases. And we believe this circadian rhythm, normally it resides in our hypothalamus. We call suprachiasmatic nucleus, SCN. So here in the light from the eyes reaches the brain and stimulates your suprachiasmatic nucleus, and thereby it is responsible for the balance of hormones and well-being. So as per the phototherapy, we believe like psychological and physical symptom of disease are imbalance of our color energy force in our body. Like example, you see the jaundice time, your color of your eyes or skin will be changed. So that balance, when there's an imbalance, like your eyes become red, so you say immediately some infection happens, some conjunctivitis. So that's the symptoms are there, correct? So these imbalance in, like example, you had a wheel flare flame, we call, right? The inflammation, we can make out the color is changing means some disease has occurred. So these are nothing but giving a symptom for the disease, the change in the color balance. So if you are going to, let's say, balance this with appropriate color, so you can restore the normalcy. Like example, we had sometime the white patches in our skin, correct? So what do we do in that time? We try to, we feel when it will be a normal color to the skin, will be out of the illness state, isn't it? So we can make out the change of the skin color is there, the change of nail color is there, the change of eyes color is there, the change of hair color, we can make out. So, but like for example, nowadays everybody got a, the gray hairs problem. So we can make out if in a young age somebody is having a gray hair means there is some problem. So that's the color imbalance exhibits, or this gives a window like there is a problem. So what is the effect of this color on our body, or the light on our body? So these light colors affects our mind, body and spirit, isn't it? Let's say I keep you in a dark room for 30 days. So your lot of physiological, emotional, and let's say, for example, you don't wake up, you don't open your eyes, you get up late in the morning, or you sleep very late in the night. So ultimately, your light is getting affected in your body. So what will happen? You will have the high, you just check any time. If you are waking up late, your blood pressure, your cholesterol, your sugar level, everything will change. So light color affects your mind, body, and spirit. The strongest impact is on your psyche. Like we say, sometime your parents also saying, right? You get up early morning, you study by that time, so you can retrieve more. Otherwise, difficult. So even ancient healers at the time believe that the red color stimulates the mind and blood flow. Blue and white color cools the body. Like example, you are wearing sometime you are in a spiritual mode. Your color you will go for a. That's why majority of the females they want to cool down. They go for a sky blue or a light colors. They go always. When they wear the very dark, intense color like a red and all, they say that they are very vibrant. So Edwin Dwight Babbitt was there who suggested that red stimulates the blood and nerves, orange and yellow excite the neural activity, and blue has got a calming and cooling effect. Even Jonathan Ott was there from American, he was a researcher who told that light rays and colors can influence the flocculation index and alter the menstrual cycle. Earlier time, not today, long back in US also, during the menstrual cycle time, all the females they used to gather in a in a single hall and they used to have get together and nicely they will be decorating. They will use the different pink colors and all, just to make them calm effect on a body. Same way here also, now you are coming from an office or you are coming from outside, you want in your home a different lights. What is this interior concept? Because it gives you cool and calm effect. So that's how you try to change the color of the light in your room. So some in vitro studies show that the colors impact the enzymatic activity. Like example, the purple enhances the glucose oxidase. Dark violet stimulates the cholesterol. Now you must be thinking why we need to know about this. Don't worry, later at last I'll let you know how we can apply this phototherapy into our research. A study was there which revealed that the warm color like red, orange, yellow increase the heart rate, the blood pressure, respiration, your muscle contraction, autonomic nervous system arousal. Whereas the cool colors have got the opposite effect. So even the investigators also gone through the colors psychological impact, like red boosts the energy and stamina, orange enhances the creativity and optimism, yellow supports the intellect and joy. Likewise, the different effects are imposed by the colors in our body. Like even these light colors have got a significant effect on the cellular molecular level, which influence the mitochondrial RNA and DNA. So a study showed that our pineal gland produces melatonin, to which we call as a chromobiotic also. Normally this hormone, it increases in night in our body, and thereby it binds to your MT1, MT2 receptor, thereby depress the central nervous system. At night time, its concentration is high. Let's say if you are not having this, if you have a habit of not sleeping in the night time and you are exposed to light continuously, so ultimately it will affect your central nervous system because it interacts further with the cell signaling molecules, even epidermal growth factor, and regulates the cell proliferation. If let's say you are changing the cycle, you have a habit of exposed to the light all time in the night also, so what will happen? It will affect ultimately your cell signals. So thereby your EGF and all the factors will be affected and your cell proliferation, differentiation, everything. Moreover, your melatonin affects your MAPK and ERK pathways also for the gene expression and thereby enhances your antioxidant enzyme activity and which assist further in neutralizing your free radicals. So it has also got the immunomodulatory and anti-apoptotic property. Even in the cancer treatment, normally we use this light therapy. So that means for the cancer treatment also we can do it. So melatonin levels are usually when a patient is of depression, bipolar disorder, PTSD, sleep disorder, at the time or in a winter season, melatonin level is changed. It is get modified. So making a balance of it, you can be relieved from these disorders. So that's a whole philosophy. Another hormone, apart from melatonin, we have serotonin. We call it as a neuroexcitatory, or we call it as a daytime hormone. You are making a mistake. You are sleeping in a daytime instead of a nighttime. Nighttime you are working, daytime you are sleeping. Just changing the cycle. So the light exposure will be more. So it will be affected. So normally in hallucinogenic or psychotic states, its level is low. So light exposure also impacts your cortisol secretion. Both blue and red light is known to enhance the production. One study was there in which a near infrared spectrophotometer was used, the radiations were used, and it revealed that the chromotherapy can enhance the oxygenation also to the brain and muscles. So even similarly, other studies were done in atopic dermatitis, and likewise, here are some clinical applications. Actually, one scientist for there, Charles Cloche, so he documented around 123 types of diseases treatment with this light therapy. There are some studies done on the adults also, like in case of anorexia, insomnia, jet lag, seasonal affective disorder, chronic alcoholism, drug addiction, anxiety, phobias, panic attacks, depression, aggression, diabetes, hypertension, immune disorder, migraine, hyperacidity, ulcers, spondylitis, hyperthyroidism, and breast cancer. All these, in these diseases, it is used as a complementary treatment. So we say the red light aids in wound healing and relieving the constipation, whereas it has got a inhibitory effect on the pathogens. And this has got a potential benefit in treating the dengue fever, severe infection, cutaneous leishmaniasis, hepatitis B, likewise many diseases. Even it is this phototherapy is known to be effective in stress relief, in reducing the depression in head and neck cancer, generalized anxiety disorder. Reports suggest that purple color, this chromotherapy, significantly reduces the hallucinations. Even in dentistry also, we are using this chromotherapy, the light therapy. We are in the patient, showing less anxiety who undergoes the anhydronic treatment with this chromotherapy. Even in auricular chromotherapy, we call it as a non-invasive treatment in which we use the colorful lights on ear to treat various health conditions. That has also shown the promising results in many psychological problems. So one physician was there who combined the auricular chromotherapy with eye movement desensitization and thereby alleviated the chronic pain. So likewise, studies also reported that your red and green color can improve the conditions, you improve the cognitive skills in your older adults compared to the white light exposure. So likewise, we have the different kind of treatment, even anti-inflammatory medications, when combined with the chromotherapy, are reported to be more effective in managing osteoarthritis when used alone. Even this light therapy has been combined with the other therapy also. You can pair with the aromatherapy, the infrared saunas, the olfactory and auditory stimulation, the magnetotherapy, the silvotherapy. That's a very common nowadays, the forest bathing, they say. We have the studies also on the children. Like example, just now I told one Iranian study was there when, anyhow, in our daily regular hospital, you see for the children, they exposed to the radiations. So we believe it plays an important role in diagnosing, preventing, and treating the disease in children also. So like using the sunlight, blue light or full spectrum white light is a well-established treatment for neonatal jaundice. That's why, if you remember your, our grandparents, when we all were the small baby, by the time they used to expose all of us to the sunlight, isn't it? They will put full the oil ointment, they will use the massage, they will do, and they will expose in the sunlight. Why? To remove all the skin problems, and you will have less, you will have more immunity will be enhanced. Your vitamin D will be more. So, so it has got I mean, this is we are using since very long time this light exposure, and they were not keeping you in the daytime like 2 o'clock or 3 o'clock in the sunlight. They were exposing mainly in the morning 8 o'clock, around 8, 9, likewise, because they were knowing that they had to give not very intense radiation. So, likewise, in reducing anxiety, in psychological distress, and many other problems, we are using your light therapy, like atopic dermatitis. Now, in this is, I told a simple way, like we all agree. Definitely, you all will agree with me. Light therapy work. That's why in many of our pharmacology models also, for zebrafish and all, many of the insect studies and all, rodent studies, we use the light experiment nowadays. Correct. So because it has got an effect. So what are the advances in this light therapy? Now we come with a technique called as photobiomodulation. So this is also called as the low level light therapy. So in this therapy, non-ionizing forms of light sources, including the lasers or LEDs or broadband lights, are used in the visible and near infrared region. And why we use it? To stimulate our cells. Basically, it's a non-thermal process. And all these devices are FDA approved also. So no issue in using these devices while designing your study. Maybe I'll come on it later. Light, normally we know that light is absorbed by the photoreceptor in mitochondria and cell membrane. So what happens nextly? It will activate the ATP in mitochondria production. And this ATP increased level activates the signaling pathway further to promote the cell proliferation, migration, and differentiation. So in this therapy, photobiomodulation, what do we do? So here, normally we keep a source near or in contact with the skin and allow your light therapy or photons to pass through the tissue. And thereby, after passes through the tissue, it interacts with the chromophore that are inside the cell. And thereby, it brings the photophysical and photochemical changes, which ultimately affects all the molecular, cellular, and tissue level of the body. And with this, a complex change of some physiological reaction occurs in your disease and damaged tissue, which causes wound healing and tissue regeneration or increased circulation, reduce inflammation. Likewise, many effects it has been given. So this is how your photobiomodulation work. Then next advancement is your photodynamic therapy. So in this, what we are doing is we are using the light source definitely, and we take a photosensitizing drug to kill the abnormal cells. So thereby we can use this photodynamic therapy for the skin condition or eye condition or some type of cancer. So it's two-stage treatment that combines your light energy together with a drug, and thereby you're using for killing the abnormal cells called photosensitizer. So this photosensitizer kills the cancerous and precancerous cell after activation from the light. That's LED normally we are using. So how this photodynamic therapy works? So firstly, what we do in this technique, we take the photosensitizing drug and will apply to the area which you want to treat. First, you will apply it. Second is your LED or your laser will be applied to activate the drug. So first you are applying the drug on the body, then thereby you are activating the drug. This activated drug reacts with the oxygen to damage the nearby cells. So this is the main principle on which it works. So where we are using this in case of skin condition like basal cell carcinoma, solar keratosis, in eye condition, so where you can treat your wet age-related macular degeneration. Then in case of cancer, you can use. So it has been reported to treat some type of cancer that include the lung cancer, esophageal, and non-melanoma skin cancer. So what are the benefits of this photodynamic therapy? It treats the small area of tissue without surgery. So you need not use surgery in the given case. And it will be less disfiguring than your surgery. Then another level is your gene therapy we use. It is the advanced technique, latest one. So here, this gene therapy is a potential treatment in case of color blindness, basically. So what we do normally in this, you will take the retina. In that, the retina, we know that it's a thin layer at the back of eye, right? So it gets detached from the surrounding blood vessel by spraying with a saline solution. Later, your missing gene will be injected into the light-sensing cells of the retina, where it will produce the protein and repair the damaged cells. So this retina reattaches again itself within a day itself. So University of Washington was there. They developed a gene therapy to restore the gene that codes for missing or faulty light-sensitive pigment. So it allows your cone cell to detect the colors that they could not detect previously. So this technique is currently being tested in the animals and have the cone cells sensitive to wavelength to blue and green light. But these are insensitive to those for the red light. So this is about your advanced technique. One more is there. The latest one, which is commonly nowadays people are using, hydrochromotherapy. So this is also another type of color therapy that uses the visible light to treat the water. And this process involves exposing water to EMR in the visible range of ingestion. So in this, you can see the bathtub will be there. So you will be putting the water inside, and you'll put the light colors different for different treatments. Then any safety, because whenever we are giving anything testing on our body, we are worried about our safety and limitations, right? So yes, your light therapy is much safer and well tolerated, although it does not affect your emotions, behavior, and cognition, but this effect may vary in quality and quantity sometime. So even the individual choices or liking also for a particular colors also affects the results. So if that person doesn't like the color, the choices or the liking is not there, then definitely that effect will not come. Coming to your light therapy conclusion, what is the use of this light therapy for all of us? Like in pharmacy, how we can, incorporate. So actually, this is very beneficial as we had discussed in mental and physical health, and especially the stress reductions. It is commendable. Reports are there and very easy to administer. Before your adoption, large scale studies are done, then proper research. If we, let's say, if we validate your research properly, if you do it properly, so then you can enhance further its role. Already one is like a jaundice treatment with the blue light, we are doing it. So the scope, how we apply this, the scope for this phototherapy is very vast. So like if you are working on stress, depression, aggression, anxiety, blood pressure, all these things, so you can collaborate with your light therapist. Like example, let's say you want to have any antidepressant drug, you are doing some experiment. Let's say you're making a formulation, or you are trying this molecule in some depression, other cases. So there you add this light component also, because there you need not take any approval. Already FDA approved, as I told, some instruments are there, so no issue in that. So it's a very easy way to do the study, because we all had a problem today to get the novelty in research. Do the same research, add this light component along with this, and do a comparison. That's a novel research for you. Correct. So then you can apply for some research grants. So it's a entirely new avenue for the research. Now coming to your one more very important aspect I was very interested in giving to you is orthomolecular medicine. Like how we see the light therapy is natural to our body. We do our treatment in a natural way. We have your orthomolecular medicine also. This orthomolecular medicine, in ortho means, that's a Greek word, right. And ortho, that's a right molecule. Orthomolecule, this is also a branch of this NCCIH. Like example, as I told, the 5 domains. So it comes under third domain, that is biological based therapies. Okay. So this concept of orthomolecular, like example for light therapy, Niels Finson, who received the Nobel Prize. Like here, this was conceptualized by the Nobel laureate Linus Pauling. So he got the 2 times Nobel Award for his research. Okay. So what is the intent of this orthomolecular medicine? And the main objective is to restore the optimal environment of body. Means it acts, that is correct the imbalances or deficiency based on the individual biochemistry. How it corrects? By using the substances that are natural to our body. So for the treatment, it will use what is natural to your body. Like example, vitamins, these are natural to your body. Amino acids, natural to your body. Hormones, natural. Metabolic intermediaries, Q10, like natural. Minerals, all natural. So with this, you can make out what is natural to your body, you're giving back. One biggest example is, for orthomolecular medicine is insulin. Your body needs insulin, you are giving back. So coming to your history of orthomolecular medicine, it's very recent science. Like in case of light therapy, it is a very old, actually very ancient, but Neil Finson became the pioneer just by inventing, be the first one to work on it. So in 20th century, these vitamins were postulated that they were used for curing the disease. 30s time, they were giving in the large dose. 40s time, they came to know like these are used in a variety of disease in a high dose for various illness such as the polio. In 60 time, 1960, the Pauling first time introduced the word orthomolecules to express the right molecules in right amount for our body. So it was the first article which came in 1968 about orthomolecular psychiatry. So coming to your concept of orthomolecular medicine, it is resting on a concept of bio-individuality. Means every person is unique. They have highly individualized nutritional requirements. So based on the difference in the anatomy, metabolism, and body composition, this bio-individuality is also differentiating. That means one single way cannot eat for everyone. Like what is good for me cannot be good for others. Like my dose of intaking the sugar is high. For me, it's okay, controlled. But for the other person, it can become a poison. So everybody's requirement of the body is different. Everybody physiology is different. So as per your orthomolecular medicine, the disease in our body can be regulated or prevented by increasing or decreasing the concentration of natural molecules like vitamins, amino acid, hormones, minerals, or metabolic intermediaries. So we can control the disease by making a balance of these right molecule or orthomolecules. Like for example, for diabetes, you need insulin, you give insulin. So it is orthomolecule. Like phenylketonuria. So you can manage it by reducing the high phenylalanine rich diet. Caesar depending on vitamin B6. So managed by giving a high dose of B6. Okay. So likewise, some examples are there. Now, what is the dosage for this orthomolecular medicine? You see, the dosage for this is not uniformized. There is no single dose for everyone. So in fact, nobody knows the exact dose for anyone. So we had to titrate the dose. Like in case of vitamin C, we are giving the high dose of vitamin C in case of scurvy patient. But for a normal person who is not scurvy, the dose will be different. So in some of the cases, sometimes we give these nutrients in a much higher dose. Sometimes in a grams also we are giving. Normally, if you see your capsules or any drugs, tablets, we give the vitamins and all in milligram, microgram. But in some cases where there's the need is high, we give in grams also. So one famous orthomolecularist, Hoffer, was there, who told like primitive people do not consume processed food, so they do not have degenerative diseases. Like why nowadays we have degenerative disorders? Because we are lacking of the essential nutrients. On the right hand side, you can see one image. We are always involved in eating these stuff. But on the left hand side, we don't take. By right, we should take it in raw form or maybe the steamed one. But don't go for totally frying and. So this is the main culprit of all degenerative diseases. You see, our grandparents, anybody's, they were surviving until 90s or hundreds or sometimes crossing the 100 also. They had a less degenerative disease. They were not having specs. So until their death also, they were doing the cycling, all that. So degenerative process was slow there because they were having full of the nutrition. Like they were having the, they were not having refrigerator in their time. The biggest culprit, you will believe, you will agree with me, like biggest culprit today is your refrigerator because we can preserve the food in our refrigerator. So again, we go for reheating. We go for fried rice, fried noodle, all that. Steaming, heating, already done once. So you are continuously heating again and again. That causes your cancer. Sometimes people feel, oh, I am taking a totally nutritious diet. I don't eat outside food. I take all the measures. But no, you got a refrigerator. I don't say that don't keep refrigerator, but yes, try to eat the fresh food. Don't go for a processed food as possible. Okay, that's why in the Western countries now the people go for the roughage. They go for salads and all more because they want to preserve all the nutrients. That's why if vegetable you're not able to consume, at least take fruits in your diet more. So some evidences are there. People sometimes they think like orthomolecular medicine is a mega vitamin therapy. So they think sometimes if we give the more of the vitamins or the nutrients in our diet, so it may cause some harmful effect. No, because these are already the natural part of your body. Okay, so it cannot be harmful. Plus, a study was there when Dr. Abraham Hoffer was there. He treated more than 10,000 schizophrenic cases in his life. And normally all of them recurred with the nutrients and vitamins itself. But there were no reported death with this. But with the other medications, yes, reported deaths are there. So people ask sometimes whether it's a placebo or not, whether simply we are eating so many vitamins and all. No, there's a response. Like example, one study was there where 25 mg of zinc was given to the people who were having antidepressant. This one, so it's their depression was reduced more than the antidepressant drug alone. When they were giving the alone drug, the response was not so good. But when they give together with the zinc as a this metal or the mineral, you say, the response was wonderful. So normally the nutritional deficiency affect the brain function in every developmental stage of life. So as I told just now, people say is like taking too much of vitamin could be harmful. No, hardly anybody would have seen any death only because of vitamin. Depression, I think this is the biggest case with which we all are going through. Normally you must be hearing in your home, in your family, relative, hey, he is a depression case, all time depressed. What is the reason behind this depression? There could be some biochemical factors. There could be some physical factors. There could be a death of somebody, the loved one, some disability, lack of social support, accidental loss of job, loss of personal dream goals. So it happens. We all had a dream and all that, And because of work stress, the depression we get. So there are many medications also that causes depression. So all these are the side effects of these medications. Then we have some genetic factors also, because of which depression may happen, or some other factors are also there. Actually, we all think, you would have seen, whenever you have done a lot of work, the physical or mental or any kind of stress, you go for, okay, let's have a drink, let's have a tea, coffee, all that, and you take a full sugar in that, some ice cream juices, all that, and you feel your depression will be down. No, you are inviting further your depression. Like, because overconsumption of sugar and processed food further exasperate your depression, like your chocolates, your pastries, breakfast, all these cookies, all these leaches your essential nutrients from your body and removes the proteins, and thereby leads to negative mood, thoughts, and nightmares. Same way, like vitamin A is there, which is a cofactor in metabolism of essential fatty acids. So a meta-analytic study was there. So that revealed that higher dietary consumption of vitamin A causes a reduction in the risk of depression. So if you eat more of the vitamin A in your diet, it will reduce your depression risk. So, and studies have shown that majority of the depression cases are there. Normally, their vitamin A level will be low. So, likewise, we have the vitamin B, your B1, B3, B6, B9, B12, all the vitamins are there. So, like it is your, for thiamine, we call the B1. So, it was been observed like normally the psychiatric patient which have been admitted to the hospital, usually they have the thiamine B1 level is low. So, how you can control? The one way is by replenishing your thiamine B1 level. Similarly, vitamin B2 is there. So a study was done in a Korean study only. It was in 2025. It was done where they showed that the high riboflavin intake reduces your 17% of suicidal ideation. Similarly, many reports are there. I'll not go in much detail. I'll come on it later, like your B3, B5, B6, B7, B8, B9. Likewise, all are many diseases they have been implicated and many study reports have been given. Likewise, we have the choline, we have the vitamin C. Vitamin C, you would have seen during the corona time, people started consuming a lot of vitamin C at the time. Why? Because research says when you give vitamin C in a higher dose, I mean in a grams and all, it has got a strong antiviral activity. So, and that time it was a coronavirus time. So everybody started consuming a lot of vitamin C and the results were wonderful at the time. Knowingly, unknowingly they have done, but that fact was being suggested by orthomolecularist. Same way, vitamin D, patients suffering from your seasonal affective disorder, usually they have a low vitamin D. That's why from the Western countries and all the people, normally they come to the Asian countries, especially for this vitamin D. Likewise, the vitamin E, essential fatty acids. So all these are the different kind of nutrients we have in our body that our body needs. You see the structure of chlorophyll and your this what do we call hemoglobin, almost similar structure, only one magnesium and iron difference is there. So that shows the similarity. So that's why in old time also they used to say take the green leafy vegetables. So that's why, but don't process it too much. Don't cook it on a very high temperature. Steamed is better, the green leafy vegetable, so that your essential nutrients will be maintained. Likewise, the zinc, copper, magnesium, whatever is needed for your body, we give. Now, all this coming to the next point. These all are, I talk about what are the key nutrients what we use in normal orthomolecular medicine practice. So what is the key principle in orthomolecular medicine? First of all, you need to have a knowledge about your safety and efficacy of it. Simply, you cannot give anybody. You need to have a knowledge. So because if you don't have, you want to design some study, better please include one orthomolecularist in your study. Second concept is these orthomolecules offer low risk of toxicity in body compared to the other conventional drug. Why? Because these are already part of your body. This is what your body needs. Third is reliability. Normally in orthomolecular medicine, the dose titration is the most practical aspect. So they do the dose titration. They keep changing the dose from lower to higher, higher to lower. They keep the change according to your disease. Then another is biochemical individuality. A dose of 500 mg cannot be uniform for all. You have to, because somebody, a disease case, you need to have a higher dose. Somebody is a non-disease state or a less disease state will require a different dose. And dietary allowance. This RDA is not normally, followed by the orthomolecularist, because this RDA is given for a healthy person, not for a sick person. So they don't rely, orthomolecularist doesn't rely on this dietary RDA report. Then environmental pollution. This is the only study in which we diagnose the toxic pollutant level also. That is also analyzed in each case. So some important aspects in orthomolecular medicine therapy is here only natural substances are used that are natural or right molecules to your body, which are the part of the body in true sense. Okay, like example, just now I was talking about like a vitamin C case, you can relieve your this antiviral effect will be there. So what are the indication where we can apply this orthomolecular medicine? In case of schizophrenia, like your ascorbic acid we are giving, or niacinamide you are giving. In case of depression, you can give your, the tryptophan, serotonin precursor we call, tyrosine, niacinamide. In case of dementia, vitamin B12. In case of congestive heart failure, CoQ10 enzyme, taurine, magnesium. In case of osteoarthritis, you can use the niacinamide, glucosamine sulfate. In case of gingivitis, that's we call as a gum inflammation or the bad breath, you can use the folic acid, CoQ10, vitamin B12, potassium, magnesium aspartate. Then in case of kidney stone, you can use the magnesium hydroxide, vitamin B6, osteoporosis, magnesium, vitamin K, and some trace minerals. Then you have your many more indications like In case of acne vulgaris, alcoholism, amyotrophic lateral anxiety, AIDS, attention deficit hyperactivity disorder. So likewise, many diseases are there. So now we think how we can apply this orthomolecular medicine from the research point of view. You see, we are using all the modern medicine, whatever you are doing, you do it, no issue. Side by side, you take one orthomolecule and do a comparative study. Plus, for a model, you can take a light experiment also. Together, you can design the three experiment. One is your modern medicine. One is your, of course, your this orthomolecular medicine in a titration dose. And one is your light therapy individually and in combo. Means together, you can do the trials for different indications, and you can apply for various grants, and you can work in the field of biochemistry, physiology, pharmacology, clinical research. Even in the formulations also you can go for, like you are from Sutix. So for all these ortho molecules, you can design some different formulations, and you can do your study. So it will be an entirely novel area in that case. Even with the orthomolecularist, you can go for direct trials also using the light therapy along with this. So that's all from my side. So these are the references. Thank you. Thank you so much for patient hearing. That's all from my side. Thank you.
Thank you, Professor Floria, for such a knowledgeable lecture. And I think every student and staff member have enjoyed that. So that was overall about your health, your life. You can implement the ideas given by Professor Floria. And I welcome any question from the audience.
Hello.
Yeah.
Hello. Hello.
Yes. You
Hello,
can go for.
yes.
Yeah.
Yeah.
Florege, thank you for excellent presentation, and it was very insightful, and I felt that it is very essential. Even common public also, not only the students or the professionals, but
Correct.
about nutrition and healthcare. Because of these days, packaged food is not a trending that much expected to the other developed countries, but still the wealthy families can afford the packaged food and the chocolates and all those biscuits and all those. It's becoming very common these days. It's like, and
True.
even the once upon time the Parle-G was only popular at the just
I'll tell
biscuit.
you, sir, even, because I think you are still there. So even in agriculture also, the plants also, people are doing the light experiments on the plants, their effects, they are studying on all the ingredients.
Yeah.
Studies are going, a lot of studies are there.
Sir, can we implement in the wellness centers this light effect? Like, I know in
Yeah,
like
why not? Why not? That's what I'm saying. Actually, if you see, normally we all are applying here. Everybody's house, you go different color shades and all. It's a effect. In fact, for this, we don't need any the approval also, because already the natural light we are using, and we can study their response.
I see, great. One more additional thought about this is there are different light sources which like in infrared is the
Yeah,
better.
that is also we are near infrared we are using in this. Nowadays in the modern advanced techniques we are using even near infrared also.
Okay,
And but normal the range we go for 372 for this visible region we are going, because that is until near infrared they had approved. Yes, it is there. We are applying.
I see. So I think in near infrared light bulbs can we distribute across the families because they
Sorry, come
can
again. Can you bit louder? Sorry.
those kind of bulbs can we introduce to for the like personal care they can put into their
No,
like
that's
bedrooms.
because in this, whatever the machines, I mean, whatever the experiment we are doing, so if that is approved by FDA, those only, because in that we have to go with the data, what is the approved one, that only we can go for. Otherwise, for other, if you're going a trial,
But
then
infrared
we have to
lights
take a
has
prior
not
approval in
not
the
not
study.
not not necessarily any regulation, I would say, because of this is
Yes.
most safest
That's what I'm
light.
saying. So we have to see in which range you are using. So we
Yeah.
can do it. Yes, we can do all the
Okay.
colorful.
Because when I went to some business fair meetings, they introduced me and they gifted me as well. But because of the plugin. I not yet
Yes,
used, but
papers are there. People are doing study on that also. Yes, they are
yeah,
doing.
yeah,
Yeah.
a very interesting talk. We will discuss
Yeah,
later.
it's actually, to be very honest, for all pharmacy, we people, normally we work in only one direction.
Yeah, yeah,
That is always, either synthesis or some phytoplants or for some, the some other biomaterial we are using and we are doing nano formulation, whatever, all AI, all that. But these are the two streams. Orthomolecular medicine is only 50-year-old or 58 or 60-year-old science. That's all. So here the chances are, because the studies are very less done. So you can do many good research, many good grants you can receive. Together with the light therapy, if we do, we'll have a different combo. I mean, you
yeah.
can totally come up with a new study, new kind of, research.
Agree. So I am also associated with the Korean cosmetic formulators and industrialists. They are very much interested about our herbal formulations
Correct.
to integrate into their cosmetic applications.
Yes.
And I think we can talk about that possible collaborations
Yeah, definitely. We can plan
and even
sometime
joint
some.
joint joint ventures as well, because we
Yes,
have to
yes, we
do
can do
some
it.
non-academic,
Correct.
like
Correct.
side by side together,
Correct. Correct.
industry academia together. Yeah.
Correct. Definitely. Definitely.
Yeah, thanks, Dr. Fullaria. It's a nice presentation. Is a new era. One can concentrate on the research, particularly pharmacy. Nowadays, the diversified field we should try to give solution.
True.
Nice.
True, sir.
You are concentrating on those areas.
Thank you,
Thank
sir.
you.
Thank you, sir. Thank you so much, sir.
Thank you, Professor Floria, for your time and a wonderful talk.
Thank you,
Everybody
Daya sir.
enjoyed that, and we will hope for more such type of talk in future APP conferences. So, uh, thank you very much.
Thank you. Thank you
So,
so much, Daya sir.
colleagues and students, we terminate our lecture sessions here. And 1:15 we will join for the oration session, e-oration competition, and rest of the activity of the hosting college will continue as per schedule. Thank you very much, everybody, for your time and listening to our speakers, and hope all the talks objectives will be clear to all of you, and any research idea you got, you will be able to implement in your future research project.
Dr.
Thank you.
Dayaji, I think award function is 3:30 p.m. Huh?
Yeah. At 1:15 we have oration competition, and in continuation, 3:30 onward we have the award
Okay. So
ceremony.
I may join in Veldict if possible, right?
Okay.
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 Thank
Thank you.
you.
Thank you. Thank you,
Welcome.
Rajesh ji.
Thank you,
राजेश जी
sir.
नमस्ते.
हाँ सर, मिलते हैं.
हाँ जी खूब
Okay.
शुक्रिया आपका.
Okay. Bye bye.
Yeah, thank you, Daya sir. Thank you
Bye.
so much.
चलिए.